These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos at my website. There's a link to my website in the annotation. The address is www.freelance-teacher.com/videos.htm. Or you can just use the link in the annotation. Thank you. In biology, there are four main types of large biological molecules. There's four types of large biological molecules. Oftentimes these are called macromolecules. Can you remember what's one of the types of large biological molecules?
Macromolecules. Disaccharide.
Good. Except disaccharide is a subtype. What type of molecule is a disaccharide? And notice a disaccharide is not that large anyway, but
it's a glucose molecule.
It could be 2 glucose. What category are those in though? Those are, what I was going for is carbohydrates.
Oh, okay. Oh, the four main ones.
That's right. So this is important because this is how the nutrition chapter is organized. It's organized around the digestion of the four main types of biological molecules. So one main type of biological molecule is carbohydrates. So let's talk about that a little bit. So one thing to keep in mind is that most of these macromolecules are what are called polymers.
Yes.
What is a polymer? A polymer is made up of a bunch of repeating subunits. So here I've drawn a polymer. Now each of these subunits is called a monomer. Do you see why those are logical names? Do you know what the prefix mono means?
One.
Yeah, and do you know what poly means?
More than one?
Yeah, it means many.
Yeah.
So these are easy names to remember. Monomer is one subunit. A polymer is made of many monomers. So how many monomers does this polymer have?
1, 2, 3, 6.
That's right. Now, in actuality, many of the polymers have maybe thousands of monomers, but I'm not going to draw that out. Yeah. Okay. Now, these monomers might be identical to each other, or they might just be similar. So they don't have to all be the same thing, but they could be similar types of molecules. So, for example, a carbohydrate is a polymer. So what's another name for a carbohydrate polymer? Carbohydrate polymer is also called a polysaccharide. Why is that a logical name? What's the poly mean?
More than one?
Yeah, there's more than one monomer in there. Okay. Now, what's the name for a carbohydrate monomer?
Monosaccharide.
There you go. Carbohydrate. Are called monosaccharides. Now, many carbohydrates are considered sugars. Many small carbohydrates, I should say. Many small carbohydrates are considered sugars. Sugars oftentimes have a certain suffix, which is -ose. actually I should just say small carbohydrates are sugars. Basically, mono and disaccharides are sugars.
Mm-hmm.
What's a disaccharide?
It's two monomer.
Two monosaccharides connected to each other, right? So monosaccharides and disaccharides are sugars. Did I mention already what's the suffix for most sugars? Words that describe sugars end with what letters? Remember, enzymes end with ASE.
Ose.
Yes.
Ose.
Sugar names. I think maybe all sugar names end in ose.
Mm-hmm.
Okay, so what's the most important monosaccharide? That would be glucose. There's other monosaccharides like fructose. The most important one is glucose.
Most important, okay, monosaccharide.
So glucose is a particularly important monosaccharide to be familiar with. I think you probably must have studied cellular respiration and the Krebs cycle in the first semester. If you recall, that's kind of based around the processing of glucose. So that's one reason I would say it's particularly important. Okay, so glucose is a monosaccharide. Let's see. What are some of the important long carbohydrate polymers? Some of the important long carbohydrate polymers. One important long carbohydrate polymer is glycogen. Glycogen is made up of many repeating glucoses.
Okay.
And it's used for energy storage in animals. Another important carbohydrate polymer is starch. It's also made of many repeating glucose monomers, and it's used for energy storage in plants. Of course, remember that many of these words are important in nutrition. So, for example, if you eat a plant, one of the advantages of eating plants is that it gives you access to their starch. And you can, the plant was using the starch for energy. Well, if you digest the starch, you can use it for your energy. And then another important carbohydrate polymer is cellulose. Cellulose is also made of many repeating glucose monomers. And it's also used by plants. Cellulose is part of the structure of the plant cell wall.
Okay.
So all three of these are large polymers, and they're all made of glucose.
Cellulose part of the structure of the plant cell.
These are all large polymers. They're all made of glucose. They're just made of glucose that's attached in somewhat different ways. We don't need to go into the details. Okay, so these are some key facts about carbohydrates. Does this make sense so far?
Yes.
Okay, good. So how is this important for digestion? So what is involved in digestion? In digestion, you eat your food, which contains many polymers. The food, in order to be digested, the food has to be broken down into monomers. So digestion involves breaking polymers into monomers so that they can then be absorbed into the body. Digestion involves breaking polymers into monomers so they can then be absorbed into the body. So, for example, if you eat an animal, it will have glycogen. Well, your digestive system has to break the glycogen down into glucose, and then the glucose is absorbed into the body. Or similarly, if you eat a plant, that will give you access to the plant's starch. Well, what happens when you digest the starch? That's broken down into what?
The starch is broken down to energy storage.
Well, its role is energy storage in the plant. But what is the starch made of? Remember, all of these are polymers of glucose.
Okay.
So if you digest starch into monomers, that would mean you're breaking it down into glucose. And then what happens to the glucose? The glucose is then absorbed into your body.
Okay. So the starch is what's made up of glucose.
Yes.
Okay.
Glycogen is a large polymer that's made of glucose. Starch is another large polymer that's made of glucose. And cellulose is another large polymer that's made of glucose.
Okay.
So when you eat a plant that has starch in it, what happens to the starch in your digestive system?
Becomes glucose.
And then it's absorbed into the body. Your digestive enzymes break it up. Good. And what happens to glycogen in the digestive system?
Turns into glucose.
It's digested into glucose, which is then absorbed into the body. Good. By the way, what happens to cellulose? Well, you do not have enzymes for digesting cellulose.
Yeah.
So cellulose cannot be digested. Cellulose passes right through the digestive system, and then it's eliminated in the feces. So cellulose is actually not digested or absorbed. That's something that's covered in this chapter. So animals can digest glycogen and starch, but animals generally cannot directly digest cellulose. Humans can't digest cellulose at all. Okay, very good. So what does the digestive system do? Maybe we should review some of this is a good time to review the basics. There are, I guess, 4 steps in digestion. Well, how is this put in the textbook? The 4 main stages of food processing. 4 steps. Four main stages of food processing. So that's one of the learning objectives for this chapter. Define and compare the 4 main stages of food processing. Do you happen to have those memorized? Do you know what those stages are?
The 4 main stages of food process. Was that be no. Glycogen? No.
Let me help you with that. So first,
Yes,
there's
please.
ingestion, which is a fancy word for eating, I guess. Then there's digestion. Then there's absorption. Then there's elimination.
Okay.
Okay, so that's something that might be a question on the exam. What are the 4 main stages of food processing?
Ingestion, digestion, absorption, and elimination.
Do you see how these are logical? Ingestion basically just means putting stuff into your mouth and swallowing it. So we're mainly focusing on food processing in mammals and humans right now. So ingestion would be putting stuff in your mouth and swallowing it. Then what is digestion? Digestion is when the polymers are broken down into monomers. Digestion is when your body breaks the polymers into monomers. That's what happens in your stomach and small intestine. This mainly happens in your stomach and small intestine.
Okay, wait 1 second. Okay.
Then what happens to the monomers after they're created? Then they're absorbed into your body. So the monomers that were created in the stomach and small intestine are then absorbed into your body in the small intestine.
Or absorbed where again? Small intestine?
Yeah, maybe I'll make a note of this. So. Okay, so this is an outline of where these occur in humans, which is the main example that this chapter is focusing on, and other mammals. Most mammals, anyway, or humans. We're mainly focusing on humans here. So ingestion occurs in the mouth. That's when you eat something and swallow it. Some digestion occurs in the mouth. Then more digestion occurs in the stomach and small intestine. What is digestion? Digestion is when the polymers are broken down into monomers. Then what happens to the monomers?
The monomers are made in digestion. They're absorbed in the small intestine.
Yeah, so the monomers are then absorbed into the body. So they're absorbed out of the small intestine and into your bloodstream and lymphatic system. The nutrients there are absorbed out of the small intestine. Some of the nutrients are absorbed into the bloodstream directly, and some of the nutrients are absorbed into the lymphatic system.
Was that in okay?
Which is another system of fluid vessels in your body. One system of vessels that carries fluid in your body is the blood vessels. Another system of vessels that carries fluid is the lymphatic system.
Mm-hmm.
So the monomers are. What happens in digestion? When you're ready.
Okay. Could you repeat that one more time?
What happens in digestion?
Oh. 1 second. I'm sorry. In digestion, the monomers, no.
Mm-hmm. Yeah, go ahead.
Sorry. The monomers, when are ingested, they are absorbed into the small intestine and into the
looks like we're getting confused. Let's take our time. So the question is, what happens in digestion? Polymers are broken into monomers. Does that make sense? Why that would be called digestion?
Yes.
Okay, we're basically taking the food nutrients and breaking them down into smaller pieces. For example, polysaccharides would be broken into monosaccharides.
Okay.
So to review, what happens in digestion?
One moment, this screen just
Okay.
Okay. Could you repeat that one more time?
What happens in digestion?
In digestion, the polymers are broken down into monomers.
Good. Then what happens to the monomers?
The monomers made in digestion are then absorbed out of the small intestine and into the bloodstream, either directly and in the lymphatic system.
Good. That's right. Okay. However, there are certain elements that never get absorbed. The things that don't get absorbed remember, for example, we talked about cellulose, right? We talked about how cellulose is not digested. And since it's not digested, it can't be absorbed. So that will pass out of the small intestine into the large intestine. And there, all those elements that were not absorbed are formed into the feces, and then that's eliminated through the anus. So that would be the last step, elimination. So elimination is when the elements that were not absorbed are eliminated from the body. Mm-hmm. Okay. So to review, what happens in digestion?
Polymers are broken down to monomers.
Good. And where does that occur in humans?
The large intestine. Small intestine. My apologies. Small intestine.
So remember, we're talking about digestion. Oh, yeah. So let's try again. What happens in digestion?
Polymers are broken down into monomers in the small intestine.
Actually, no.
Oh.
Digestion occurs in the mouth, stomach, and small intestine.
Okay. Okay.
Digestion occurs in the mouth, stomach, and small intestine. Then their absorption occurs in the small intestine, and then elimination through the anus and the large intestine.
Okay, food.
So perhaps take a few seconds to kind of study this diagram and tell me when you're feeling comfortable with it.
Okay, so digestion polymers are broken down to monomers from the mouth, stomach, small intestine. Monomers. Okay. All right.
Okay. So what happens in digestion?
In digestion, the polymers are broken down into monomers.
Okay, good. And then what happens to the monomers? And where does digestion occur in humans?
It happens in the mouth, then the stomach, and then the small intestine.
Yeah. And what happens then to the monomers?
Monomers made in digestion are absorbed into the absorbed out the small intestine and then into the bloodstream, either directly and in the lymphatic system.
Yeah, so the monomers are either absorbed into the bloodstream or into the lymphatic system. Good. And then what happens to the food elements that were not absorbed?
The food elements that are not broken down are eliminated to the large intestine and then the anus.
Right, as the feces. Okay, very good. All right, so that's our basic process here. So food is taken in humans. We'll look at the human digestive system. It's taken in the mouth. What happens to the food after you swallow it? Do you know where it goes next?
It goes into the stomach.
Good. There's a tube, though, that connects the stomach and the mouth.
Do
you know what the name of the tube is that connects the mouth and the stomach?
I mean, I would say throat, but
So the throat is the beginning of that tube. That's right. The throat, which is the pharynx, is the beginning of that tube. But the tube itself, from beginning to end, is called the esophagus.
Okay, all right.
That's a term that's likely to be important on the test. So the tube that connects the mouth and the stomach is the esophagus. So you enter into the esophagus through the pharynx. The pharynx is at the beginning of the esophagus, but is just one part, or the beginning, or the entrance to the esophagus.
So should I have this diagram memorized too?
Yeah. And you were asking me, yeah, so this diagram we're making is definitely a good diagram to memorize. So one of the learning objectives for this chapter is describe the structural components of the mammalian digestive system. So that's kind of what we're doing here. We're describing the structural components of the mammalian digestive system. The first component is the mouth. Then the next component is the esophagus. Then the next component is the stomach. Okay, good. So continuing. Do you know where does the food go? Where does the food matter enter after it leaves the stomach? Do you know what the next part of this is all called the digestive canal or the alimentary canal or the digestive tract. So do you know what's the next step after the stomach?
The small intestine.
That's right. Very good. And do you know what's the next step after the small intestine?
The large intestine.
Obviously, this is just a cartoon. It doesn't show the real shapes. If you've ever seen a real picture, you know that the small intestine and the large intestine are highly coiled around each other. This is just to give you a cartoon that shows the basic order that things pass through. All right, and then so as the food matter is passing through the large intestine, it's being formed into the feces. The feces is then stored at the end of the large intestine, which is called the rectum, and then it's eliminated through the anus. Okay, so this is just a broad overview of the most important elements of the digestive system in humans, but most other mammals are somewhat similar to this. Okay, so we definitely need to know this order. Most of this I think you already knew. What's the name of the tube that connects the mouth and the stomach?
The esophagus.
Good. That would be a good thing to know. Okay, and as we were discussing here, what's the function of the mouth? Well, obviously the mouth is where the food enters, but we also mentioned that there's some digestion that occurs here. So I was saying, we saw earlier that digestion of some things begins in the mouth.
Okay.
All right, what about the stomach? Does the stomach do digestion?
It breaks down the food.
So it does do more digestion.
Yes, basically.
Does the stomach do absorption?
No.
No, that's certainly not one of its main functions. There's not much absorption in the stomach. How about the small intestine? Does the small intestine do digestion?
Yes.
And does it do absorption?
Yes.
Yeah. So most absorption occurs in the small intestine. What's the function of the large intestine? We haven't talked about this yet, but the large intestine has two main functions. One is absorption of water.
Okay.
A lot of water is absorbed in the small intestine too, but more then there's more water absorbed in the large intestine, and also the formation of the feces to prepare for elimination. These two things go together. Okay, so this is just we don't have much time left, so this is just a very broad overview of the key elements of the digestive system. Okay, so this would be a good cartoon to be familiar with for the exam. So going back to our large biological molecules, so what happens to glycogen when you eat it? Do you remember what is glycogen made up of? You can see again from the screen. What's glycogen made of?
Glycogen is made up of glucose.
That's right. So what happens when glycogen is digested? What happens to glycogen when it's digested?
It's created into glucose.
That's right. And then what happens to the glucose?
It's stored as fat.
Well, that would be taking us further than I wanted to talk about today.
Okay,
the point is, what happens after the glucose is digested? It's absorbed.
Okay.
That's my point. Then it's absorbed into the body. Okay, good. So, that would be some important points then about carbohydrate digestion. Let's see. Okay, so that's a broad overview of carbohydrates. So remember, we're talking about the four types of large, maybe we'll try to add one more thing here. So carbohydrate digestion occurs in the mouth and the small intestine.
Mm-hmm. The only function of the mouth is for digestion and, chewing up the food.
That's right. So obviously it functions in ingestion, bringing in the food, then chewing, but there's no major absorption occurring in the mouth. So digestion of carbohydrates occurs in the mouth and the small intestine, as we put in our picture here. So where does digestion of carbohydrates occur?
In the mouth and the stomach.
Take your time.
And the small intestine.
Now, not in the stomach. The stomach does digestion, but not of carbohydrates. Do you see how I've written this out here? The carbohydrate digestion occurs in the mouth and in the small intestine. Notice I didn't write carbohydrates next to the stomach. So the stomach does digestion, but not of carbohydrates.
Okay.
The stomach does digestion, but not of carbohydrates.
All right. The stomach does digestion. Okay.
So where are carbohydrates digested?
The small intestine.
And where else?
In the mouth. In the mouth.
That's right. Do you see how I'm trying to write that out in the picture here?
Mm-hmm.
Carbohydrates are digested in the mouth and in the small intestine, and then they're absorbed by the small intestine. Okay, good. So where are carbohydrates digested?
In the mouth and the small intestine.
So we said there's 4 main types of large biological molecules. We've only talked about one so far. What's the one type of large biological molecules that we've talked about so far?
Carbohydrate.
Carbohydrate, that's right. What type of molecule, what class would glucose fall into? That's a carbohydrate. What class does starch fall into? That's a carbohydrate. What class does glycogen fall into?
Carbohydrate.
Good.
No, I mean, yeah, it's a glucose. It's made from glucose.
So it's a carbohydrate. And what class does cellulose fall into?
Carbohydrate.
Okay, good. Can you think of any of the other large types of biological molecules besides carbohydrates?
Lipids.
Excellent. So what are the types of lipids? The types of lipids are steroids, phospholipids, which are important in cell membranes, and fat. The one we're focusing on for digestion is fat. So in this chapter, we're focusing on fat. Notice how many of these names in digestion are important in nutrition. Obviously, carbohydrates are an important source of nutrition. Fat is an important element in studying nutrition. So here we're focusing on fats. What these have in common is that they're all not soluble in water. These are not soluble or only have parts that are soluble in water. Okay, so here we're talking about fat. Here's what a fat molecule looks like. So fat molecules look like this. Notice fat molecules are not polymers. Do you see why I would say that? They're not really made up of repeating subunits. Fat molecules are not polymers. So when this is digested, it's broken down into this. This little cartoon is representing glycerol. And these are fatty acids. These are just cartoons to kind of represent the vague kind of shapes that these have. So notice that one fat molecule is digested into 1 molecule of glucose and 3 fatty acids. One fat molecule would be digested into 1 molecule of glycerol and 3 fatty acids. Good.
Could you, is that all you could scroll up, or were there some words on the top?
Can you see now?
Yes, thank you.
Tell me when you're ready.
Okay.
Okay. So do you see how this is not a polymer? It doesn't have a structure like this. It's not just one monomer after another. So it's not a polymer, but it's still considered one of the main types of large biological molecules. So to go back to the main work that we're doing on here, we'd like to know, where is fat digested? So it turns out that fat is digested only in the small intestine. Fat is digested only in the small intestine. So where is fat digested?
In the small intestine.
What happens to fat when it's digested? it's broken into glycerol and fatty acids. And then what happens to the glycerol and fatty acids? What's the next step after digestion?
Oh, it's absorbed into the...
By the small intestine.
Yeah.
It's absorbed in the small intestine. Good. So fat would then be absorbed into the small intestine, by the small intestine. Good. So where is fat digested?
Fat is digested in the small intestine.
Good. And where is it absorbed?
The small intestine.
Good. Where are carbohydrates digested?
Carbohydrates are digested in the mouth and the small intestine.
And then what happens after they're digested?
After they're digested, they're absorbed in either... they're absorbed...
Notice, really, the only place that nutrients are absorbed is the small intestine.
Yeah.
All the large intestine does is mainly absorb water. So all of the nutrients are absorbed only by the small intestine. Sorry, you were saying?
Yeah. No, yeah, I was just agreeing with you.
Okay, good. Okay. And by the way, carbohydrates are digested into monomers. What's the name of a carbohydrate monomer?
Monosaccharide?
That's right. So the carbohydrates are digested into monosaccharides, which are then absorbed by the small intestine. Fat is digested into glycerol and fatty acids, and that's absorbed also by the small intestine. So we're just trying to go over the most important parts here for digestion. Okay, so what's one of the major types of large biological molecules we've talked about so far?
Lipids.
Lipids. And we've only focused on one main type of lipid, which is fat. That's the only one type that's important in the digestion chapter. Mm-hmm. All right, and what's the other type of large biological molecule that you and I have talked about so far?
Carbohydrate.
Yeah, good. And what's the name of a carbohydrate monomer?
Monosaccharide.
Very good. And what's the name of a carbohydrate polymer?
Polysaccharide.
There you go. Can you remember a third type of large biological molecule?
Nucleic acid.
Very good. There are two important examples of nucleic acids. What are the two types of nucleic acids?
I remember. No, I actually don't remember.
One of them is, sorry?
Amino acid?
Actually, that's separate. One of them is DNA.
Okay, yeah.
And what's the other one?
RNA?
That's right. Do you know what the Na stands for in DNA? It stands for nucleic acid.
Oh.
And what does this Na stand for?
Nucleic acid.
As well. Okay. So these are the two types of nucleic acids that are important in biology. Good. Nucleic acids are also polymers. So these are the nucleic acid polymers. Two types of nucleic acid polymers are DNA and RNA. Now, in the first semester, you studied what the monomers are called here. You might have forgotten that. Do you happen to remember what are the names of the nucleic acid monomers? I might have forgotten. Those are called nucleotides.
Oh, yeah. Yes, yes.
Do you remember that actually the DNA code looks like this? Adenine, guanine, thymine, cytosine, and then they're paired up like this? Do you remember this from the first semester?
Yes, I do.
These are the nucleotides. Adenine nucleotide, guanine nucleotide, thymine nucleotide, cytosine nucleotide. RNA also has a uracil nucleotide. So A, G, C, T, U, those are the monomers. Okay, so anyway, these are the monomers here are nucleotides. Okay, so when DNA is digested, it would be digested into nucleotides, and then actually the nucleotides are digested even further. The nucleotides then get digested even further, and then they're absorbed. The digestion chapter actually doesn't spend much time on that, so we won't spend much time on that either. I'll just briefly add that to our picture. If I can find my picture. Here's my picture. Okay. So nucleic acids are digested only in the small intestine. And there's only one organ that absorbs, that plays a major role in absorption. What's the one organ that plays the major role in absorption?
Small intestine.
That's right. The large intestine mainly is just absorption of water.
Mm-hmm.
Okay, good. Okay, this digestion chapter didn't pay too much attention to nucleic acids, so we'll just say that and then move on. Okay, good. So what's one of the main large types of biological molecules we've talked about so far?
Carbohydrates.
Carbohydrates, that's right. What's the name of a carbohydrate polysaccharide?
Oh, hello?
Hi, can you hear me? Testing,
yeah,
testing.
It's back. It just had...
So
okay,
what's the name of the carbohydrate polymer? What do carbohydrate polymers called?
Polysaccharide.
That's right. And where are polysaccharides digested? Which organs do polysaccharide digestion? Which organs do carbohydrate digestion?
In the mouth and the small intestine.
Mouth and small intestine. Good. So they're digested into what? What are the carbohydrate monomers?
Monosaccharide.
Monosaccharides. Good. What happens to the monosaccharides after they're digested?
The monosaccharides. Excuse me. They're absorbed into out the small intestine and into the bloodstream or the lymphatic system.
That's right. Maybe I'll just mention. Might as well be complete. Carbohydrates and nucleic acids are absorbed into the blood out of the small intestine. Fat is absorbed into the lymphatic system. Fat is the only nutrient that's absorbed into the lymphatic system. The only nutrient that's absorbed into the lymphatic system is fat. Everything else is absorbed into the bloodstream. Okay. You have that in your notes?
1 second.
Sure.
Okay.
Good. Okay, so one type of large biological molecule is carbohydrates. What's another type of large biological molecule besides carbohydrates?
The ones that we discussed are a new one.
Ones we've discussed.
Lipids.
Lipids. What's the main type of lipid we focus on in the digestion chapter?
Fat.
Fat. That's right. Where is fat digested?
Fat is digested in the small intestine.
Right. Notice that that's the only place I've written fat in this picture. And remember, it's digested into glycerol and fatty acids. What happens to the glycerol and fatty acids after they're digested in the small intestine?
I'm sorry, could you repeat that?
What happens to the glycerol and the fatty acids after the fat has been digested?
Oh, the glycerol, It's well, it's it's digested only in the small intestine. It's absorbed by the small intestine.
Yeah, it's absorbed out of the small intestine, and that's the one nutrient that's absorbed into the lymphatic system.
Yes.
Good. And we also talked about nucleic acids. They're also digested in the small intestine and then absorbed out of the small intestine into the bloodstream. Okay. There's one more type of large biological molecule. We've talked about carbohydrates, we've talked about lipids, we've talked about nucleic acids. Can you remember what's the fourth type of large biological molecule?
Amino acid.
Good, except those are small. What's the name of the large molecule that's made of amino acids?
Amino?
Protein.
Oh, geez, protein, yeah.
Okay, and obviously that's an important type of molecule in your bio class. Okay, proteins. Another name for proteins is peptides. So a protein polymer is called, it could just be called protein, or it could be called a polypeptide, or maybe just a peptide. So protein polymers are called proteins or polypeptides.
Okay, proteins, polymer. Okay.
When you're ready, do you know what the name of the protein monomers are?
Monopeptides?
That would definitely be logical, but there's another very specific name. It's a name you've already mentioned today.
Nope.
What are the protein monomers called? What are proteins made of?
Geez. I mentioned that?
Yeah, these are the amino acids.
Okay.
Proteins are made of amino acids. So remember that a polymer...
Monomer. Protein.
So a protein looks like this. Here would be one amino acid, then another amino acid, then another amino acid, then another amino acid. The names of the amino acids are things like lysine, threonine, histidine, phenylalanine, tryptophan. So this could be tryptophan, lysine, phenylalanine, aspartic acid, maybe another phenylalanine. So there's 20 different amino acids. So a protein is made up of a string of amino acids.
Okay.
Okay, so what's the name of a protein monomer? What's the name of a protein monomer?
Amino acid.
Yeah, I think that's pretty important. All right, so the protein monomers are the amino acids. Good. Proteins play many different roles in the bodies. There's many different types of molecules that are proteins. What's one of the most important types of molecules that are proteins? It's a type of molecule we've talked about today. What's one of the most important roles that proteins play in the body? It's kind of a vague question, but any idea what I'm going for?
Like the function?
Yeah, what's one type of molecule that's a protein?
Hold on.
That was kind of a weird, vague question. What I was going for is, what type of molecule is an enzyme?
Okay. All right.
So what type of molecule is an enzyme?
Protein?
That's right. Okay. So that's one of the, there's many types of protein. Proteins can be enzymes. They can also be receptors. They can also be ion channels. Do you remember we talked about the calcium ion channel earlier? I suppose that would be a protein. There's lots and lots of other roles that proteins can play that I'm not thinking of right now. So there's many roles that proteins can play in the body, but one of the ones that's especially to focus on is that they're enzymes. Okay, good. And by the way, remember, how can you tell if how can you tell if something is naming an enzyme? What do enzymes usually end in?
ACE.
They usually, not always, but they often end in ACE. Good. There's many other proteins that don't end in ACE, however. Okay, very good. So what's the name of a protein monomer?
Amino acid.
Yeah, and what's another name for a protein polymer besides protein?
Polypeptide.
Yeah, polypeptide. Okay, good. So protein digestion begins. You should be able to figure out where it begins because we've come to the end of the molecules and we haven't used the stomach yet. So that must be what the stomach is for. Protein digestion begins in the stomach.
Okay.
Whoops. And then protein digestion continues in the small intestine.
So, okay. Protein digestion starts in the stomach and begins in the small intestine?
Oh, yeah.
It begins in the stomach and continues in the small intestine. All I'm saying is some protein digestion occurs in the stomach, some protein digestion occurs in the small intestine.
All right.
So notice the mouth only digests one type of molecule. What's the only type of molecule that the mouth digests?
Carbohydrates.
And the stomach only digests one type of molecule. What's the only thing the stomach digests? When you're ready, the stomach only digests one type of molecule. What's the only type of molecule the stomach digests?
I said protein.
Oh, I'm sorry, you cut out. Okay.
Oh, okay.
And then notice the small intestine digests all types of molecules.
Mm-hmm.
So what are the 4 types of molecules the small intestine would digest?
Carbohydrates, fat, nucleic acids, and protein.
That's right. On the other hand, the mouth only digests one thing. What's that?
Carbohydrates.
And the stomach only digests one thing, which is?
Protein.
So now that we see the pattern, this shouldn't be too hard to memorize. The small intestine digests everything. The mouth digests one type of molecule, and the stomach digests one type of molecule. What's the other function of the small intestine besides digestion?
Absorption.
That's right. It absorbs the monomers from all this digestion.
Mm-hmm.
Okay, good. And then the large intestine is mainly only for absorption of water. Okay, good. All right, good. So what would be one of the main types of biological molecules? Oh, and then we may as well finish this all off here. Remember that where does absorption occur?
Absorption happens in the small intestine.
Right. Remember that the only thing that's absorbed into the lymphatic system is fat. So carbohydrates, nucleic acids, and now we can add proteins are all absorbed into the blood. Actually, I guess I shouldn't call these, well, they're not, their monomers are absorbed, not the polymers. So carbohydrate, nucleic acid, and protein monomers are absorbed into the blood. Okay. So, when you're ready, what's one of the main types of large biological molecules?
Carbohydrates.
Yes. What do we call a carbohydrate polymer?
Polysaccharide.
Excellent. Where does carbohydrate digestion occur?
In the mouth and the small intestine.
Excellent. So the carbohydrates are digested into what?
Carbohydrates are digested into glucose.
Good. And what's the name of carbohydrate monomers?
Like glycogen, starch, cellulose.
Those actually are not monomers. So remember, the polymer is called a polysaccharide. What is the name of the carbohydrate monomers in general?
What
I was going for was monosaccharides.
Oh, wow.
Glucose
Okay,
is
yeah.
only one type of monosaccharide.
Yeah.
It's maybe the
I
most
pro-
important, but it's only one type. So what would we call a carbohydrate polymer?
Polysaccharide.
And that's digested into the monomers, which are called
Monosaccharide.
including glucose. Good. And then what happens to those monosaccharides after they're produced?
They're broken down into.
Well, they've already been broken down.
Yeah.
And then what happens after they're produced?
They're absorbed.
In which part of the digestive tract?
Small intestine.
Yeah, absorbed into the bloodstream. Good. So what's another type of large biological molecule besides carbohydrates?
Lipids.
Good. What's the main type of lipid we focus on in the digestion chapter?
Fat.
Fat. Good. Where is fat digested?
Fat digested in the small intestine.
Good. Remember that each fat molecule is broken up into glycerol and fatty acids. And then what happens to the glycerol and fatty acids?
It's absorbed into the lymphatic system.
Out of the small intestine. Excellent. Good. So those are not, fat is unusual because it's not a polymer. What's another type of large biological molecule? These are also called macromolecules. So what's another type of biological macromolecule? Besides
Nucleic
carbohydrate
acid.
and fat? What were you saying?
Nucleic acid.
Nucleic acids. That's right. What are the two types of nucleic acid polymer?
DNA and RNA.
That's right. What does the Na stand for in DNA?
Nucleic acid.
That's right. What's the name of the nucleic acid monomers?
Nucleotides.
That's right. And where are nucleic acids digested?
In the small intestine.
That's right. And then what happens after they're digested?
Oh, they're absorbed into the blood.
That's right. So the substances that are formed from their digestion are then absorbed into the bloodstream. Okay, good. So we've talked about carbohydrates, fat, lipids, and nucleic acids. What's the last type of biological macromolecule?
Proteins.
Proteins. That's right. What's another name for a protein polymer?
Polypeptides.
Polypeptides.
Oh.
That's right. Good.
Right.
And where are proteins digested?
Proteins are digested in the stomach and the small intestine.
Good. Remember that the small intestine digests all the main classes. Good. When a protein is digested, what's it broken into?
Wait. It's broken down into. Ooh.
What's the name of a protein monomer?
Oh, they're broken down to amino acids.
That's right. Because what's the name of a protein monomer? Amino acids.
Amino acids.
Good.
Yeah.
And then what happens to the amino acids?
They're digested in the stomach.
Well, that's actually a mistake. They're produced by digestion, right? The amino acids are created in digestion, and then what happens to the amino acids?
Oh. And I don't.
What I was going for is that the protein is digested. What's the product of protein digestion?
Amino acid.
Right. So notice, it doesn't make sense to say the amino acids are digested. They're formed by digestion. And then the amino acids are absorbed. That's what I was going for. What happens after digestion? Absorption. Then the amino acids are absorbed. Out of the small intestine into the blood. So, do you see, it doesn't make sense to say that amino acids are digested. Do you see why I would say that?
Yeah.
Yeah.
Because they're already
the proteins are digested to form amino acids. Good. So, what's another name for a protein polymer?
Polypeptide.
Good. And what's the name of the protein monomer?
Amino acid.
Good. Okay. What's the name of a carbohydrate polymer?
Polysaccharide.
Is digested into what?
Monosaccharide.
Because those are the monomers. Good.
Mm-hmm.
And then what happens to the monosaccharides?
The monosaccharides, they're absorbed out the small intestine into the bloodstream or the lymphatic system.
They're absorbed out of the small intestine and into what?
Into the lymphatic system or bloodstream.
Good. Now remember, there's actually only one type of nutrient that enters the lymphatic system. Do you remember the only type that goes directly into the lymphatic system? That was the fats.
Okay. Oh, okay.
We're going over a lot of stuff pretty quickly because we're running out of time. But anyway, the fats are absorbed into the lymphatic system. Everything else is absorbed into the blood. So monosaccharides are absorbed out of the small intestine and into where?
Into the bloodstream.
Right. Okay, good. Where are the proteins? Where are these proteins digested?
In the stomach. And yeah. No, not in the, yes, in the stomach and in the small intestine. Small intestine digests all of them.
Right. Where are the carbohydrates digested?
In the mouth and the small intestine.
Okay, good. Fat is digested where?
Fat digested in the small intestine.
Only. Good. Remember that this is not a polymer. It breaks apart into glycerol and fatty acids. And then what happens to the glycerol and fatty acids?
Oh, 1 second. The glycerol and fatty acids, they're absorbed into the lymphatic system. Absorbed by the intestine.
They're absorbed out of which organ?
Small intestine.
And into
The lymphatic system.
Okay. Nucleic acids are not really focused on very much in the nutrition chapter, but anyway, we can say, so maybe I'm not going to write them down specifically here, but keep in mind, what's the name of a nucleic acid polymer? What are the two types of nucleic acid polymer?
Well, nucleic acid polymers are DNA and RNA.
And what are the name of their monomers?
Nucleotides.
That's right. Okay. So we talked about that a little bit before, but the nutrition chapter doesn't focus on those too much. So I'll just stop with this. These are the three main types of digestion here that that this chapter is focusing on here. Okay. Well, we tried to get through a bunch of stuff here, but this is all stuff that would certainly be helpful on the test. And so you keep in mind, we just skimmed the surface here. There's way, way more details that we didn't have time to get into today. There's all kinds of enzymes that are involved in digestion, like amylase and pepsin and stuff. There's the role of hydrochloric acid. There's the role of hormones that hopefully maybe some of that you've learned on your own. So there's a whole bunch of stuff that we didn't get to today, but I tried to go over the stuff that was most important, that might be most helpful on the test. So hopefully you have a little bit of time left. So I'll send you my notes here and you can try to study it. So this little cartoon here I think could be very useful in preparing for the test tomorrow. I think this little flow chart here I was a little bit messy, but hopefully I was clear. If you can understand what I wrote here, this could be very helpful in preparing for the test tomorrow. I think these three little flowcharts we just went over about proteins, carbohydrates, and fat, I think if you can memorize those, that could be very helpful for the test. And I think it's also very important for this chapter and for the whole rest of the course to know these basic facts about the 4 types of biological molecules: carbohydrates, lipids, nucleotides, and proteins. Like I said, nucleic acids might not really be tested too much in this chapter, but they're worth knowing because they're going to be important for the rest of the course as one of the main types of biological molecules. And in our previous session, we talked about how the beginning of the digestive tract is the mouth. So let's review, where would the food go after exiting the mouth?
Down the esophagus.
That's right. And then what's the next destination after? Oh, so let me label that. And then what's the next destination after the esophagus?
The stomach.
Good. And then?
The small intestine.
Good. And then?
The large intestine.
Okay. And then what's at the end of the chain?
Then it ends with the rectum and then the anus.
That's correct. Okay. All right, something else that we were discussing last time then were the 4 main types of large biological molecules. So can you remember what's one type of large biological molecule?
Carbohydrate.
That's right. So what would be another name for a carbohydrate polymer?
Polysaccharide.
Good. And then what would be the name of the monomers?
Monosaccharide. Oh, by the way, I spoke to my professor today. He said we're not going to need to understand he said we don't need to look in depth for the nucleic acids. That's not one of the main ones. He just wants to know just the main food groups that'll be broken down, which would be carbohydrates, lipids, and
Good.
Yeah.
Okay, so we'll review that. Okay, that's good to know. I think that's kind of how we were doing it last time. We mentioned the nucleic acids, but I think we mentioned that the chapter didn't seem to be focusing on that. But anyway, that's good to have confirmed. Okay, good. So here we have the polysaccharides and the monosaccharides. Good. What's another name for the monosaccharides? A kind of nutritional name?
Carb monomer.
So the point is that they're generally sugars.
Okay.
Mono and disaccharides are generally considered sugars. How can you recognize a sugar from its name?
It has os at the end of it?
Yes. Sugars generally end in os. For example, what's the most important monosaccharide that comes up the most in biology?
Glucose.
Right, which is a sugar. Okay, very good. We also briefly talked about 3 main types of polysaccharides. Can you remember one of the main types of large polysaccharides we talked about? We didn't talk about that as much, but do you remember one of those?
No, that's the main types of polysaccharides?
Yeah, so we talked about 3 types or groups of polysaccharides that are important.
Okay,
remember any of
glycogen?
those? Sorry, say that again, please.
Glycogen.
Excellent. That's what I was going for.
I thought it was monosaccharide.
Glucose is a monosaccharide. Glycogen is a polysaccharide. Glycogen is made out of a bunch of glucoses.
Okay.
So glycogen is one of the main molecules used for energy storage in animals. So if you eat another animal, one of the purposes of eating another animal is to get access to their energy. Well, a lot of that energy will be gotten from their glycogen. And glycogen, again, is made up of just a string of glucoses. So when glycogen is digested, it's converted just into a bunch of glucoses eventually. Okay, so that's one of those. Energy storage in animals is from glycogen. Do you remember what's the name of the compound that's responsible for energy storage in plants?
Starch.
That's right. It's similar to glycogen for plants. And again, it's made up of a string of glucoses. So if you eat some vegetables or some plants, you're hoping to get some access to some of the energy they've stored largely in their starch. And when starch is digested, it turns into what?
Glucose?
That's right. The whole purpose of digestion is to turn the polymer into the monomers. Good. Okay, and then there's one more type of polysaccharide which is used as for a structure of plant cell walls.
Cellulose?
That's right. That's also made of a string of glucoses. All three of these are made of a string of glucoses arranged in slightly different fashions. Now, if you remember, cellulose cannot be digested by animals generally. So, at least not directly. So the cellulose would not be digested. The cellulose would pass right through the digestive tract and then would be expelled again in the feces because it can't be digested by humans. And by many other animals too. Okay, so so those are some key ideas here about carbohydrate. So in general, when you eat a carbohydrate, it's digested into what?
To glucose.
Yeah, and other monosaccharides. These ones are digested into glucose. There's other types of carbohydrates that are digested into other types of monosaccharides. Good. And then what happens to the monosaccharides? They're absorbed into the body. Okay, so do you remember where does carbohydrate digestion begin?
I'm sorry, could you say that again?
Where does carbohydrate digestion begin? In which organ of the digestive tract here starts the digestion of carbohydrates?
For some reason you're breaking up. I heard what you said. It's just that in the mouth?
That's right. So we can say that carbohydrate digestion begins here. And which other organs digest carbohydrates here?
The small intestine?
Yes. Okay, good. So good. And then what happens to the carbohydrates? So the carbohydrates are digested into what?
Glucose?
And other
monosaccharides?
Yeah, that's right. And then what happens to those monosaccharides?
They are turned into glucose?
Well, that would be getting ahead of ourselves. What's the next step in the process? So in the we have a bunch of monosaccharides in the digestive tract. What happens after digestion?
They're absorbed?
Yeah, absorption. So the next step would be absorption into the body. So which of these, in which step here are the monosaccharides going to be absorbed?
In the small intestine?
That's right. That's going to occur here in the small intestine. And are the monosaccharides absorbed into the bloodstream or into the lymphatic system?
Bloodstream?
That's right. Carbohydrates are absorbed into the bloodstream. Okay, excellent. So those are some key ideas there about carbohydrates. So that's a good recollection of that. So let's add a few more details. We need to know one of the key enzymes that digests carbohydrates in the mouth. Maybe you've picked this up from class. What's the name of the key enzyme that's digesting carbohydrates in the mouth?
Amylase?
Good. And enzymes are generally secreted by glands. What are the glands that secrete the amylase into the mouth?
Oh, we did talk about it in class. Oh, I don't think we got in depth with the glands. He just mentioned that.
Okay, well, I'll give you some help with that. So this is one that would make sense to you once I mention it. It's the salivary glands. So you already know from your from common sense that salivary glands are secreting into the mouth, and the saliva that they secrete includes what's called salivary amylase. So we can make a note here. So here's a salivary glands. And they're going to be secreting salivary amylase. If you remember, we already talked about another type of amylase when we were working on plants. Do you recall we talked about how alpha amylase was involved in the stages of germination?
Yes.
Right. Do you remember we said, what did the alpha amylase digest?
Was it the endosperm?
That's right. It digested the starch, I believe, in the endosperm, and it converted it into what?
Glucose.
Yeah, well partly,
sugar.
Yeah, I guess if it was starch, it would digest it into sugar. So anyway, it's digesting the carbohydrates in the endosperm and digesting it into sugars. So that's a relative of this type of amylase, I suppose. So what type of molecule is salivary amylase? What type of molecule is this salivary amylase?
An enzyme?
That's right. It's an enzyme. How can you tell that from the name?
It has the ACE at the end of it.
That's right. And what does salivary amylase do?
It breaks down the carbs in the mouth.
Yeah, it breaks them down into smaller subunits. Okay, good. By the way, what type of molecule is an enzyme? Enzymes fall into what group of molecules?
No. Would it be amino acids?
That's not quite right, no. So it falls into the group of proteins.
Okay, all right.
Proteins are made of amino acids, but an enzyme would be way more than just one amino acid.
Yeah.
So that's an important idea. So to review, what group of molecules is salivary amylase in? Well, it's an enzyme. And what group of molecules are enzymes in?
Proteins.
That's right. All right. And what are proteins made of?
Amino acids.
So what's salivary amylase made out of?
Enzymes.
What's salivary amylase made out of? It is an enzyme. But what's it made out of?
Proteins.
It is a protein. And what are proteins made out of? Amino acids. So what's salivary amylase made out of? Amino acids too. If you see what I'm saying. Because this is a protein, it's made out of a string of amino acids. Okay. It's just important to see the relationships between these different types of molecules. Okay, and then moving down to the small intestine, we're going to have more digestion of carbohydrates. So there's a few different enzymes that are involved here. There's one in particular that I think might be tested, though. Can you think of, or remember from class, any of the enzymes that digest carbohydrates in the small intestine?
Oh, well, we talked about the enzymes in the stomach, not the small intestine.
You mean in class or in the tutoring?
In class.
Yeah, okay. So did you finish this chapter in class, or is there more left over to cover?
No, what is this chapter? What chapter is this? 41?
Chapter 41, yeah. So are you finished with that, or is there more to cover in the next lecture?
There's more to cover in class. We haven't finished yet. We finished the end of 40 today. We finished end of 40, and then we began 41, and we stopped at there was a slide there's a rabbit digesting food, and that's where we stopped.
Okay, fair enough. Okay, so in any case, some of this maybe you haven't quite yet seen in class, but we may as well go over it now in one organized fashion. So it turns out that the enzyme so my question was, remember you already told me that the small intestine is involved in digestion of carbohydrates. Well, what's one of the key enzymes involved there? It turns out that one of the key enzymes involved in the digestion of carbohydrates is going to be another type of amylase.
Okay.
Again, there's a good chance that would be tested. So this is easy to remember because it's amylase in both places. Now, who is going to be secreting that amylase into the small intestine? There must be another gland that's going to be secreting that amylase into the small intestine. And that gland turns out to be the pancreas.
Okay. So should I put like, all right, say the enzyme is secreted from the pancreas?
That's right. In fact, it'll kind of be obvious because the full name of this enzyme is pancreatic amylase.
Okay.
So if we write down that the name is pancreatic amylase, it shouldn't be too hard to remember what's the name of the gland that secretes this pancreatic amylase.
Pancreas?
There you go. That's right. Not too hard to remember. Okay. So those, I think, there's a few other enzymes involved in carbohydrate digestion, but these are the ones I think that are likely to be tested. So we'll just put these down. So it's not too hard to remember. These are both amylase. And again, I suppose they're relatives of the amylase we studied in the plant chapter, alpha amylase. Okay, so what type of molecule is pancreatic amylase?
An enzyme?
That's right. And what does it do?
It allows the stomach to break down carbs.
Yeah,
small intestine.
Yeah, it breaks down carbs, continues to break them down into smaller pieces. Okay, good. And what type of molecule is an enzyme?
A protein?
That's right. So what type of molecule is pancreatic amylase? A protein as well. Okay, good. And what's the gland that secretes the pancreatic amylase?
Pancreas?
And it's secreted into what other organ?
The small intestine.
There you go. Okay, very good. So those are some extra important points there about carbohydrate digestion. Okay, good. So those are some of the key points about carbohydrate digestion. So can you remember what's another one of the types of large biological molecules besides carbohydrates?
Lipids?
That's right, lipids. Okay. By the way, the definition of lipids is lipids are compounds that are not soluble in water. Lipids are compounds that are not soluble in water. What's the main type of lipids we focus on in the digestion chapter?
Fatty acids?
Now, fatty acids are just a part. The type of lipids we're focusing on here are fats. If you remember, you could kind of cartoon out the structure of a fat like this. So notice that the fat is not a polymer. It's not made of a bunch of repeating subunits. It's not a polymer. When the fat is digested, it could be broken down into a kind of cartoon that kind of looks like this. This compound here is what's called glycerol. And these are what are called fatty acids. So that was what you mentioned a moment ago. You had mentioned the fatty acids. Fatty acids themselves are not really a main type of lipids. They're a structural component that's used to make lipids. So notice glycerol plus 3 fatty acids makes a fat. Glycerol plus 3 fatty acids makes a fat. Okay. Okay, so let's review what we just talked about there. What do lipids all have in common? What's the definition of a lipid?
The compounds are not soluble in water.
That's right. What's the main type of lipid that we focus on in the digestion chapter?
Fats.
That's right. And when a fat is digested, it's digested into what?
Into 3 fatty acids. Into glycerol.
And glycerol, yeah. the fat is broken up into glycerol and 3 fatty acids. So it's not a polymer, so the digestion here works a little bit differently than before. Okay, very good. So do you remember from what we discussed earlier, where is the first place in this diagram where fat will be digested?
Hold on one second.
Take your time.
Small intestine.
Good. The small intestine. So we also will have digestion of fat here in the small intestine. Good. So when the fat is digested, it's broken down into what?
Glycerol.
Good. And what else?
3 fatty acids.
There you go. That's right. Okay. So now the fat is going to be a little bit trickier to talk about here. Remember, fat is a lipid. And what was our definition of lipids?
Lipids are compounds that are not soluble in water.
That's right. But that makes it difficult to digest the fat. Things can be digested most easily, so the substances that are moving through the digestive tract are moving in a watery medium. There's water throughout the digestive tract, and what we're hoping is that they're all going to be dissolved in little pieces into the water, so the digestive enzymes have lots of surface area to digest them. What happens when you put fat in water? When you put fat in water, it just globs up. If we put a bunch of fat in water, we get a big fat in water. Instead of getting a bunch of little dissolved particles that can be easily attacked by the digestive enzymes, instead we get a big, disgusting glob of fat. Well, this is too big for the digestive enzymes to digest effectively. So the first task that the digestive system has to accomplish here is they have to somehow break up this big glob into smaller droplets. We need to break this up into smaller fat droplets. Now remember, this is occurring in the small intestine, so the small intestine is going to need some substance that is able to break up a glob of fat into smaller droplets. I don't know, did you happen to pick up on that from class? What the substance is? That this is what's called, it emulsifies the fat. I don't know if your class has covered this yet. Here we have these droplets of fat here. All right, so it turns out that these are going to be created by bile salts. The bile salts are going to break up the glob of fat into smaller droplets of fat. So this is an important part of fat digestion. So we might kind of cartoon the bile salts like this. So the bile salts are going to kind of glom on to these little droplets and make it easier for them to dissolve or disaggregate in the water. Okay, so the bile salts are going to break up this big glob of fat into smaller droplets, so the droplets can then be digested. Okay, good. So where does the bile salts come from? So the bile salts come from bile, and bile is created by the liver. So let's add that into our picture. So bile is a compound that contains bile salts. This is a little bit complicated now because the liver does not send the bile directly to the small intestine. Instead, the liver sends the bile into the gallbladder.
If you don't mind, do you mind if I try to quickly sketch this image again because I'm running out of room?
Oh, on your paper? Yeah, sure.
Yeah.
Yeah, take your time. Since I can't see you, sometimes I kind of start running ahead of where the student is. So anytime I do that, it's fine to just tell me you need a little bit more time to take notes. So go ahead and take your time and tell me when you're ready to keep going.
I apologize. 1 second. Almost finished.
That's okay. Take your time. It's important to have, the whole purpose of the tutoring is to just give you some clear notes to study. So take your time and make sure that you have it down clearly.
Okay. All right.
Okay, ready to keep going?
Mm-hmm.
All right. So how does the liver aid digestion? What does the liver do?
It creates bile.
And then what does the gallbladder do?
The gallbladder, it stores it.
Yeah. So the liver makes bile and sends it to the gallbladder until it's needed. And then where does the gallbladder send the bile? Into the small intestine, because that's where the digestion of the fats is going to happen. By the way, you know what a bladder is? A bladder is just kind of a biological bag or container. For example, when people say the word bladder, they usually think of the urinary bladder, which is a bag that stores urine. I think that gall is just an old-fashioned name for bile. So when we say gallbladder, we mean a bag that stores gall or bile. So the purpose of the I think the only purpose of the gallbladder is to store the gall, or what's called now the bile. Okay, just like the urinary bladder stores urine. Okay, so that was created by the liver. All right, so what does the liver do?
Creates the bile.
And where does the bile go then?
It's stored in the gallbladder.
And then what?
The gallbladder will send the bile into the small intestine.
When? Well, when there's food here that has fats in it, hopefully, that need to be digested. Okay, notice that this is efficient This way, it would be pointless to send the bile into the small intestine when there's no food or no fat there. Then it would just be wasted, and you'd have to make some more. So it makes sense to make it and then store it for when it's needed and then secrete it in here. Okay. So, and then what does the bile do? How does the bile aid the digestion of fat?
Because the bile salts created breaks down the giant glob of fat into smaller droplets.
Yeah, into smaller globs, basically. Good. So we take the big glob and break it into smaller globs. Okay, good. So that was this picture. That was this picture here. Okay, and then what's going to happen to those droplets of fat? Well, now these are small enough that they can be worked on by digestive enzymes. So we'll need some enzymes here to digest it. Remember that the enzyme that digested carbohydrates was called amylase. So what's going to be the enzyme that digests fat? Well, it turns out that it's called lipase. So the lipase is going to digest the fats. So first, the bile salts break down the big glob into smaller globs of fat. That's called emulsifying them. And then the lipase actually digests the fat droplets. And remember, after fat is digested, when fat is digested, it's broken into what pieces?
Wait, I'm sorry, could you say that again?
When fat is digested, it's broken into what sub-pieces?
Droplets.
Now, after those are digested, those are broken down into what? Remember, what is one fat molecule made out of? We talked about this a little bit earlier. Each fat molecule is made out of glycerol plus 3 fatty acids. So at the end of digestion here, the lipase will be digesting the fat into glycerol and fatty acids. Okay. And then, do you remember from previously, the glycerol and fatty acids are absorbed into where?
They are absorbed into the lymphatic system.
Excellent. That's good that you recalled that. They're absorbed into the lymphatic system. Unlike all the other nutrients which are absorbed into the blood. All the other nutrients are absorbed into the blood, but the products of fats are absorbed into the lymphatic system. And where does this absorption occur?
In the small intestine. In the lymphatic system. Oh, yeah.
They're absorbed from the small intestine into the lymphatic system. They exit the small intestine and enter into the lymphatic system. Good. Okay, good. Okay, oh,
I'm sorry. Would you mind? Sorry. I didn't finish copying down the notes.
Yeah, sorry. Go ahead. Take your time.
Thank you. Is it necessarily always 3 fatty acids?
Well, if a fat molecule always looks like this. So if it's digested completely, it always does create 3 fatty acids. That's right. It's a little more complicated than that. Some of the fats are not completely digested, but I think that's more detail than you need for your course. So for your course, we can just say if it's completely digested, it would always create, when one molecule of fat is completely digested, it always creates 1 molecule of glycerol and 3 fatty acids. Okay. Yeah. Even that might be a little bit more detail than you need, though. Maybe it's good enough just to say it forms glycerol and fatty acids. That's the key.
Okay, thank you.
Sure, that's fine. Okay, so to review, in which part of the digestive tract does fat digestion occur?
In the small intestine?
Yeah, in the small intestine. So now remember that fats are lipids. What's the definition of lipids? What do all lipids have in common?
Lipids are compounds that can't be broken down in water.
Did you say they can or cannot?
They cannot.
Right, they're not soluble in water. So originally you just have a big glob of fat in the small intestine. Then what?
So when there's a big glob of fat, the bile that is stored from the gallbladder is sent when there's food in the stomach. The bile is sent into the small intestine, and then it creates the bile salts, which will allow the bile salts will break down the large globs of fat, and then they will attach to the smaller droplets, and then the lipase will digest the fats.
Okay, good. Yes. All right, so we have a big glob of fat. So what we need is we need bile salts that are contained in bile. Where is the bile produced?
In the liver.
Good. And then where does it go after the liver?
The gallbladder.
Good. And then after the gallbladder?
The gallbladder sends it to the small intestine.
Right. And then what the bile salts do is they start coating the outside of the big glob. And by coating the outside of the big glob, they aid the big glob to break up into little pieces that are more soluble in water. That's basically what you said. That's called emulsification. So that's basically what you said. They coat. So it's a little bit complicated. The gallbladder is secreting a fluid called bile, and one of the components of bile is the bile salts. And the bile salts are going to coat the big glob and help it break up into smaller globs. Then that will allow the lipase to digest the fat droplets. Okay, good. By the way, now we have to say, where does the lipase come from? I don't think I mentioned that before. The lipase comes from the pancreas. In fact, sometimes this is called pancreatic lipase. Let's see, maybe we'll add that to our table here. So we have digestion of fat by lipase. So the pancreas we have here, we've seen that the pancreas secretes pancreatic amylase, and now we've seen that it also secretes lipase. Okay. Good. And the lipase is going to be digesting the fat droplets. So who secretes the lipase?
The pancreas.
That's right. It secretes it into where?
It secretes into these. No. Into it.
It does secrete it into the small intestine, because that's where the lipase is needed to digest the fats. So that's kind of what I'm trying to show in this picture here. Notice that both the gallbladder and the pancreas secrete into the small intestine.
Okay.
Both the gallbladder and the pancreas, they both secrete into the small intestine.
Okay.
Okay. So who secretes the lipase?
The pancreas.
Into where?
Small intestine.
Good. What type of molecule is lipase?
Enzyme.
That's right. And what does the lipase do?
It breaks down. It digests the fat droplets.
Good. Into what?
Glycerol and fatty acids.
Okay, very good. That's right. Why did these have, by the way, why did these have to be broken into smaller globules before the lipase could digest them? Well, this is just kind of common sense. The enzyme can only digest the surface of the glob, right? Because it doesn't have access to the interior. So if this was a lipase, the lipase could only digest around the surface of this glob because the interior is not accessible to it. So what we need is to increase the surface area. The more surface area there is, the more places that the lipase can attach. And do you agree that if you take, say, a big glob, and then say we break it into two pieces, these two pieces now have more surface area, right?
Yes.
Because now we have this additional surface area here. This additional surface area that didn't exist before. And then if you break those up again, now there's tons more surface area than there was before. Here, 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 here. Tons and tons of surfaces that didn't exist before. So the key is, the reason we have to break the glob into smaller droplets is that the more you break it up, the more surface area there is. And the more surface area there is, the more places that the lipase can attack and start digesting. What would happen if there was not enough surface area? If there wasn't enough surface area, then most of the fat wouldn't get digested by the time it had passed out of the small intestine. And then it would just be wasted and excreted in the feces. So we need to be able to digest this in a reasonable, efficient and timely fashion before it passes out of the digestive system. So the idea here is that surface area, increasing the surface area, is necessary to increase the digestion. Okay, so then the lipase creates glycerol and fatty acids, and what happens to the glycerol and fatty acids?
The glycerol and fatty acids, they're absorbed into the small intestine and
out of the small intestine.
and then into the lymphatic system.
That's right. Okay, so to review again, fats are what type of biological molecule?
They are lipids.
And what's the definition of a lipid?
A lipid is a compound that is not soluble in water.
That's right. So where are fats digested?
The fats are digested in the small intestine.
That's right. But originally in the small intestine, they would just be a big glob because fats are not soluble in water. So what happens in the small intestine to digest them?
So in the small intestine, the liver, which creates the bile, is going to no, the liver creates the bile. The gallbladder is what stores the bile.
Right.
And the gallbladder will secrete, when there's food in the small intestine, it'll secrete the bile into the small intestine, which allows, I guess, it allows it to break down the big globs of fat into smaller droplets.
That's right. So the bile contains bile salts. And the bile salts break down or emulsify the big glob into smaller globs. That's right. Good.
After that, they coat the large glob of fat, and that breaks down into the smaller droplets. The lipase, which comes from the pancreas, will digest the fat droplets, and then they're created into glycerol and fatty acids, and they're absorbed out of the small intestine into the lymphatic system.
Okay, excellent. Good. Very good. By the way, how do bile salts break down the big glob into smaller globs? How does emulsification work? That's actually, it's not too complicated. That's something you would study if you take introductory biochemistry or maybe organic chemistry. I don't think we have to go into that for biology. So we're not really going into how the bile salts do this. We're not going to explain how they break down the big glob. But it's not too complicated. It's something you would see in maybe an OChem or a biochem class. Okay, good. So very good. And let's see, we also were talking about carbohydrates. So where does carbohydrate digestion begin?
In the mouth.
That's right. What's the key enzyme that does the carbohydrate digestion in the mouse?
Salivary amylase.
Good. Which comes from what gland?
The salivary gland.
Obviously. Very good. And then where else are carbohydrates digested?
In the small intestine.
That's right. By what key enzyme?
Pancreatic amylase.
That's right, which comes from what gland?
Pancreas.
Like lipase does. Okay, good. And when carbohydrates are digested completely, they become what?
Oh, when
Carbohydrates are digested into what?
glucose.
Glucose is only one example. What's the name of a carbohydrate monomer?
Monosaccharide.
So they're digested into monosaccharides. That's right. And then where are the monosaccharides absorbed out of?
It's not the lymphatics. Bloodstream.
Into the bloodstream. That's right. They're absorbed into the bloodstream from where?
Wait, into the bloodstream from the small intestine?
From the small intestine. That's right. Okay, good. All right, so that's some good progress on those. So we've talked about carbohydrates and we've talked about fats. What's another of the types of large biological molecules?
Protein.
That's right. What's another name for a protein polymer besides protein?
Polypeptides.
Right. And what do we call protein monomers?
Amino acids.
Right. That's a very important term. So a protein is just a bunch of amino acids all linked up to each other. Okay, very good. What's one? Proteins play a bunch of different important roles in the body. What's one important type of protein molecule? What's one very important type of role that proteins play?
Being like enzymes.
Yeah.
Proteins, they can.
So that's what I was going for.
Yeah.
Enzymes. They play many other roles. They're antibodies. They're receptors. They form some hormones.
Ion channels.
Excuse me?
Ion channels.
Ion channels, excellent. Yeah, many different roles. Enzymes is just one of the many important roles that proteins play. How can you recognize when something is an enzyme from its name?
Usually have ACE at the end of it.
Right. Not always, but often.
Yeah.
Like lipase or amylase. Okay, good. Okay, so those are some key ideas about proteins. So we talked in our previous session, where does protein digestion begin?
Oh, 1 second. The stomach.
That's right. We haven't talked much about the stomach yet because doesn't play a key role in carbohydrate or fat digestion, but it's important in protein digestion. All right, so the stomach plays a key role in the digestion of proteins. Good. And there's a key enzyme that's released in the stomach. We haven't talked about this yet. Do you happen to know it?
Pepsin.
There you go. That's right. So the key enzyme here, digestion of protein by pepsin. Good. So what type of molecule is pepsin?
An enzyme.
That's right. It doesn't end in ase, but it's still an enzyme. Good. And what type of molecule is an enzyme?
Protein.
That's right. So pepsin is a type of enzyme, which is a type of protein. All right. And what does pepsin do? It digests proteins, or it begins the digestion of proteins. Very good. And where else are proteins digested besides the stomach?
The small intestine.
So notice the stomach digests everything.
I mean the small intestine.
Yeah. Did I misspeak? So yeah, the small intestine digests all three of these types, all the types of nutrients. So this also digests proteins. Okay. Digestion of proteins continues here. So notice, digestion of proteins occurs here in the stomach. And there's a bunch of different enzymes that would be involved in that. I'm not sure if it can be tested on their names though, so maybe I won't write them down. Again, a bunch of the enzymes that are involved in this would be secreted by the pancreas.
Are not all the enzymes, but like the main ones just for digestion, or would usually be secreted by the pancreas.
So, so first of all, the pancreas secretes into where?
Into the small intestine.
Yeah. So, yeah, the enzymes that work in the small intestine come from two places. The enzymes that work in the small intestine come from two places. Some of them come from the pancreas, just like you were saying.
Mm-hmm.
And some of them just come from the border of the small intestine, what's called the epithelium of the small intestine. If you studied that term in class, remember that the epithelium of an organ is kind of the layer of cells around its surface or its border. So the border of the small intestine actually secretes a lot of enzymes straight into the interior of the small intestine. We haven't talked about that much. So the enzymes that work in the small intestine, some of the enzymes from the small intestine were created in the pancreas, and some of them were created in the epithelium of the small intestine.
Okay.
And so those are the two sources. The
Right.
the enzymes that work in the small intestine come from two sources. Some of them come from the pancreas, and some come from the epithelium of the small intestine.
Okay.
Okay. Your textbook goes over a bunch of other names of enzymes, but I don't want to go into more detail than you're going to be tested on. So for now, I think I'm not going to put down any more names. The names I put down are the ones that are most likely to be tested, I think. But it is good to know that the pancreas is secreting some of the enzymes. So where do the enzymes that go into the small intestine come from? Some of them come from the pancreas, and where do the other enzymes in the small intestine come from?
Um. Oh, what you just I forgot. I forgot.
From
that
the
name.
epithelium? Is that a term you've seen before?
No.
Oh, no? Okay. So epithelial cells are cells that are at the surface or edge or border of a substance. They kind of seal the border between one organ and another organ. So basically, the outer surface or the outer border of the small intestine secretes enzymes into its interior.
Oh, the epi. I thought that was just mainly for tissue.
Let's see. I'm not quite sure if I followed you there. So let's see. I'm not sure what you mean by saying that it's for tissue. So
I mean, like,
many different organs have an outer surface that's made up of epithelial cells, and that's the case for the small intestine as well. So the small intestine is basically a hollow tube. The small intestine is a hollow tube. Inside the hollow tube, digestion occurs. But what's the surface or the border of the small intestine made out of? It's made out of a sheet of epithelial cells.
okay.
One important type of tissue is epithelial tissue, or epithelial cells is an important type of cell, and those are usually at the surface or the border of an organ. So the outside of the small intestine is made of these epithelial cells, and they also secrete enzymes into the interior of the small intestine.
Okay.
So where are the enzymes for the small intestine coming from? Some of the enzymes in the small intestine come from the pancreas. And where do the other enzymes in the small intestine come from? We were saying that the enzymes in the small intestine, some of the enzymes in the small intestine are secreted by the pancreas. And where do the other enzymes in the small intestine come from?
The epithelial cells
Of
in
the
the
small
in the
intestine.
small-
That's right, the outer surface of the small intestine. Okay, good. All right, very good. So when proteins are digested, they're broken down into what?
They're the proteins are broken down into poly polypeptides.
That
No,
would be
no,
the
no,
name
no.
of a polymer.
Yeah.
What's the name of a protein monomer?
Oh, amino acid.
That's right. So ultimately, they're broken down into amino acids.
Mm-hmm.
And then the amino acids are absorbed out of where and into where?
They're absorbed out of the small intestine into the bloodstream,
That's
blood
right.
into the blood.
That's right. Okay, good. Okay, good. So let's see. So to review these processes, there's only one type of biological molecule that's digested in the mouth. What type is digested in the mouth?
Carbohydrates.
That's right. By what enzyme?
Amylase.
Yeah.
Or yes.
Amylase from where?
The salivary amylase from the salivary glands.
That's right. Good. And then when the food exits the mouth, it enters into where?
Into the small, well, it goes down the esophagus and down into the stomach, and then
Good. So
it's
let's stop at the
digested.
stomach for a second.
Oh,
So then there's only one type of molecule that's digested in the stomach. What's the type of biological molecule that's digested in the stomach?
pepsin. Well, oh, protein is digested in
Protein,
the stomach.
yes. Protein is digested in the stomach by what type of enzyme?
Pepsin.
That's right. Okay, good. And then where does the food enter after the stomach?
The small intestine.
That's right. And what types of molecules are digested in the small intestine?
Well, all the ones we discussed.
All of them, yeah. So, for example,
For example, proteins, fats,
proteins, fats, and
carbs.
carbs. That's right. So proteins continue to be digested in the small intestine. Carbohydrates are continue to be digested in the small intestine. What enzyme did we talk about that digests carbohydrates in the small intestine?
Amylase.
Yeah, amylase. From what gland?
Pancreas.
That's right. Good. And then what happens to the proteins and the carbohydrates after they're digested?
The proteins are broken down into amino acids.
Good. And the carbohydrates are broken into?
The carbohydrates are broken into monosaccharides that become glucose, sugars.
Yeah. Now, what's the relationship between the word monosaccharide and the word glucose? Is glucose a type of monosaccharide, or is monosaccharide a type of glucose?
Monosaccharide is a type of glucose?
No, actually, glucose is a type of monosaccharide. So
Yeah.
carbohydrates are made up of many repeating subunits. Some of the subunits could be glucose, but some could be other things like fructose or ribose or galactose. So glucose is just one type of monosaccharide. We just emphasized it because it plays an important role in the biology course and in biology, but it's just one type of monosaccharide.
Mm-hmm.
Okay, so carbohydrates are digested into what?
Okay, carbohydrates are digested into well, okay. Sorry, I'm getting a little confused.
That's okay. Well, let's look at it this way. What's the name of a carbohydrate polymer?
Polysaccharide.
And what's the name of a carbohydrate monomer?
A monosaccharide.
That's right. So what are carbohydrates digested into? Monosaccharides. The point is that the reason we were talking about polymers and monomers is that when digestion is complete, things have been broken down into their monomers.
Okay.
When digestion is complete, the polymer has been broken down into the monomers.
Mm-hmm.
So what's the name of a carbohydrate polymer?
Polysaccharide.
And a carbohydrate monomer?
Monosaccharide.
And a protein polymer?
Protein polymer is polypeptide.
Yeah, or just protein. And what's a protein monomer?
And amino acid.
That's right. So what are proteins digested into?
Amino acids.
That's right. And what are carbohydrates digested into?
Monosaccharides.
That's right, including glucose. But glucose is just one type. Okay, and then what happens to the amino acids and monosaccharides after they've been formed in the small intestine?
They're absorbed out of the small intestine into the bloodstream.
Excellent. Good. All right, and something else that's digested in the small intestine is fat.
Mm-hmm.
And the fat is broken down into what?
The fat...
When fat is digested, it's turned into what?
Into glycerol and the fatty acids.
That's right, and then that's absorbed out of the small intestine into?
The lymphatic system.
Okay, good. All right, good. So we've talked about carbohydrates, lipids, and proteins. There's one more type of large biological molecule. We don't need to spend too much time on that for this chapter, but just to make sure we're organized here, what's the last type of biological molecule?
Nucleic acids.
And what are the two main types of nucleic acids?
DNA and RNA.
That's right. What does the Na stand for in DNA?
A nucleic acid.
Right. So these, the polymers are DNA and RNA. And again, we won't just spend a second more on that because it's not too important in this chapter, but what's the name of a nucleic acid monomer?
Nucleotides.
That's right. Okay. But I think you said that your TA told you that we weren't going to have to focus on that too much for the digestion chapter, so we'll just leave it at that. But these are, again, digested, I think, mainly and absorbed mainly in the small intestine. Okay, good. So let's see. Let's go back up to our picture here, and let's start adding. So we've gone over some of the key ideas here, but there's some important details. So food starts in the mouth, and then where does it go?
Down the esophagus.
That's right. However, really, to get from the mouth to the esophagus, it has to pass through your throat area, right? Do you remember what's the technical name for the throat?
Trachea?
We haven't really talked about that. What did you say?
I said trachea.
Okay, that's just a guess. Trachea turns out to be the windpipe, not the food pipe. So the throat here is your pharynx.
Oh, pharynx. Yes.
So basically, the pharynx is like the passage from the mouth into the esophagus. The pharynx is the passage that leads from the mouth into the esophagus. So there's not really much digestion happening there. It's just part of the path. Okay. How does food move down the esophagus? You might think that the food was just dragged down by gravity, but that's actually not true. You don't need gravity to move food through the digestive tract.
Yeah.
So have you heard in class, what is it that actually is moving the food through the esophagus and then through the small intestine and the stomach?
The smooth muscle?
Smooth muscle contracting, yes. Since it's smooth muscle, does that mean the contractions are voluntary or involuntary?
Involuntary?
Yeah, you don't usually think about, gee, I need to move some food through my esophagus right now, right? It just happens without you thinking about it. All right, there's a name for those contractions of smooth muscle that move the food down. So for example, if the food was right here, maybe this portion would contract and then move the food to here. And then maybe this portion would contract and move the food to here. And then maybe this portion would contract and move the food to here. So there's waves of contraction. Did you happen to pick up on the name of those contractions?
No.
So those might be on the test. That's called peristalsis.
Okay. Okay, should I write this on like a separate?
Wherever you think is organized. I'm not going to write it right here just so that I don't clutter up this diagram, basically. Because this occurs over the entire digestive tract. Food is moved down the digestive tract by waves of contractions of smooth muscle. Smooth muscle that lines the digestive tract. And what are those contractions called? Peristalsis.
Okay.
Okay. So what do we call the waves of contractions that move through food through the digestive tract?
Peristalsis.
That's right. So for example, you could still eat even if you were standing on your head. Even if you're standing on your head, the food would still be moved now up the esophagus. That would happen. It would move up the esophagus by those waves of contraction. It doesn't require gravity. Okay, good. Let's see. Another important topic to talk about here. One of the objectives for this chapter is define and compare the 4 main stages of food processing. We talked about this last time. What would be the 4 main stages of food processing?
Okay, so it'll be consumption, ingestion. One second.
And the next ones are ones that that are kind of logical that we've talked about. Obviously, you have to put the food in the digestive tract first.
So it's ingestion, and then I get.
So
I know
they're together. Word. So these are logical once we see them. After the food is ingested, it has to be digested.
Mm-hmm.
So that's the second phase.
Ingestion, digestion, absorption, and then elimination.
Right. So these are fairly logical stages. Where do these occur? Where does this occur?
In the mouth.
Good. And where does this occur?
In the stomach and small intestine.
And also in the mouth, right? Because remember that starch begins to be digested in the mouth. And then where does this occur?
In the small intestine.
That's right, pretty much in the small intestine, although there's more absorption of water in the large intestine. Mm-hmm. All right, and then this occurs.
Elimination happens in the rectum and the anus.
That's right. So the feces are prepared in the large intestine and then eliminated through the rectum and the anus. Okay, good. All right, so what are the four main stages of food processing?
Ingestion, digestion, absorption, and elimination.
Good. Okay, now to talk about this a little bit more, so basically after the food is ingested, it exists as a bunch of polymers. But in order to be brought into the body, it has to be broken down into little pieces or digested, and then it can be absorbed. Now, there's a problem here. So notice that all of these digestive enzymes here could be very dangerous to your body, right? Because what do these digestive enzymes do? They take biological molecules and they break them down. So your body has a problem. How can you be sure that you're going to be breaking down your food without breaking down your body, if you see what I'm saying? So your food is made out of carbohydrates and proteins and fat, so you want the digestive enzymes to break down your food, to break down the carbohydrates and fats and proteins. But your body is also made. Your body is also made of carbohydrates and fats and proteins. So there's a danger that these digestive enzymes could start just breaking down your body. Obviously, that would be very dangerous. So that's one of the problems that the digestive system has to face. It has to find a way to digest your food without digesting you. Does that make any sense?
Yes.
Yeah. So what's the solution? How does the digestive system accomplish that? Well, basically the solution is that the food is kept inside the digestive tract while it's being digested. And the digestive tract is, the way a biologist thinks about it, this is a little bit weird, but to a biologist, your digestive tract is outside of your body. In a way, the digestive tract is like a hollow tube that's running through your body. So the point is, all of these digestive enzymes are secreted into this hollow tube in the middle of your body, so they don't have access to the rest of your body, basically. They don't have access to the rest of your body because they're outside of your body in this hollow tube. It might seem a little weird to think that your esophagus is outside of your body, but it's kind of like a donut hole. The hole of a donut is kind of outside of the donut, right? Well, the digestive tract is just kind of like this very long, thin donut hole that's extending through the animal's body or through the human body. It's kind of a hollow tunnel that runs through the body and is, in a sense, outside of the body. So that's why these digestive enzymes are not digesting the body, because in a way they're not inside the body. They're secreted into this digestive tract, this hollow tube that runs through the center of the body, but which, in a sense, isn't really inside of the body. Okay, so that's an important perspective to have on these ideas. Now, as the food moves down, when the food reaches the stomach, we already said that there's going to be pepsin secreted into the stomach. Something else, though, that's very important that's secreted into the stomach is hydrochloric acid. The lining of the stomach secretes hydrochloric acid into the stomach. The lining of the stomach secretes hydrochloric acid into the stomach. So would that make the stomach a acidic or basic environment?
Acidic.
Because of the hydrochloric acid. That's right. Do you remember, does that mean it has a high or a low pH?
A higher pH?
So actually, an acidic environment is a low pH. Acidic means lots of protons floating around. And when there's lots of protons, that actually means you have a low pH. Even though pH has an H in it, it moves in the opposite direction to the number of protons. Lots of protons, or very acidic, means a low pH. And a basic, or alkaline, environment means a high pH.
All right.
Okay. So does acidic mean high or low pH?
Low pH.
Right. How about basic?
High pH.
Right. And did you pick up on what does alkaline mean? Alkaline means the same thing as... I didn't quite emphasize that, but alkaline is another word for basic.
Okay.
What does alkaline mean?
Basic.
So what does it tell you about the pH?
Means the pH is high.
Right. What would be a neutral pH?
Water.
That does have a neutral pH, but what number would it be? Neutral
7?
pH is 7. That's right. So let's say you had a pH of 4. Would that be acidic or basic?
Acidic.
Right. How about if you had a pH of 10?
It'll be basic.
Right. Or alkaline. Good. So the stomach, is the stomach acidic or basic?
It's acidic.
That's right. Good. What keeps it acidic? What does the stomach secrete to keep itself acidic?
The hydrochloric acid.
That's right. So would it have high or low pH?
A low pH.
In fact, it turns out the stomach's pH is around 2. That's very, very acidic. Normally, body tissues are close to 7 in pH. Normal body tissues are not far from 7 in pH. This is much more acidic.
Mm-hmm. Don't the doesn't the stomach have like a special lining around it to prevent like the tissue from decaying? Or
Exactly. That's right. So this strongly acidic environment would normally tend to break down the wall of the stomach. So the stomach is constantly replacing the epithelial cells around its exterior, and it's also constantly secreting mucus to protect the stomach lining. That's right. So remember, I was saying that the problem with digestion is how do you keep from digesting yourself? So the question is, how does the stomach keep from digesting itself? Well, the hydrochloric acid is secreted into the interior of the stomach, and the border or lining of the stomach is constantly being protected by mucus and basically reconstructed to keep it from getting broken down. But I believe if the lining of the stomach does become penetrated, that's what I think is called an ulcer. If you have an ulcer, that's when the acid or the enzymes are eating through the lining of the stomach. Okay, good. Okay, why do we need this? What's the purpose of this very acidic environment? Well, that very acidic environment helps to break things down. I think this is so acidic that you could actually break down. I think the textbook says you could break down iron nails at this pH. Wow. So it's very acidic. So obviously that's going to help us to break down the food. Also, it helps to kill any bacteria that came in with the food. Most of the bacteria are not going to be able to survive that acidic environment. Also, this enzyme, pepsin, doesn't work except in very acidic environments. This enzyme, pepsin, only works in a highly acidic environment. So that's another reason why we need that very low pH. Most enzymes work best at a pH of around 7 because that's the normal pH of the body. But pepsin is unusual. It's adapted for the stomach. So what do we call this food matter in the stomach? Once the food matter gets into the stomach, it gets churned up into this, I suppose if you could see it, it would really be a very disgusting acidic glob. And it's called chyme. So the food matter in the stomach is called chyme, or acid chyme, because it's become very acidic. Okay. Okay, so what type of molecule is digested in the stomach?
Protein.
That's right. By what key enzyme?
Pepsin.
Right. And what else is secreted into the stomach besides pepsin?
Hydrochloric acid.
That's right, which keeps the pH high or low?
Low.
No. And what do we call the food matter once it reaches the stomach?
Chyme.
Chyme. Okay. Now, remember here we have a problem. This pepsin, the pepsin is being created in the lining of the stomach. The pepsin is created in the lining of the stomach. Now, pepsin is a very strong digestive enzyme. So what is going to keep the pepsin from just digesting the lining of the stomach before it gets secreted into the interior? Well, the answer is that most of these digestive enzymes are created in an inactive form. Most of these digestive enzymes, when they're first created, are in an inactive form.
All right. Okay. I'll just put that in the note.
So the stomach's epithelium secretes pepsinogen into the stomach. And we don't need to worry about the pepsinogen eating away at the stomach epithelium before it's secreted, because the pepsinogen is inactive. It's not digestive yet. So now in the interior of the stomach, we have pepsinogen. And then also another part of the stomach epithelium secretes hydrochloric acid into the stomach. So the interior of the stomach will have both pepsinogen and hydrochloric acid. And it turns out the hydrochloric acid activates the pepsinogen. The hydrochloric acid, I think, cuts away a piece of the pepsinogen. It makes it shorter and makes it active.
Oh, what was that? What is the H plus hydrogen? Is that the sign for acid?
HCl is the formula for hydrochloric acid.
Oh, it looks like, I'm sorry.
So this is the chemical formula for hydrochloric acid, HCl.
Got it. Do you mind scrolling to the left a bit?
Sure, take your time.
Thank you. Okay. Thank you.
Okay, so the point is one part of the stomach epithelium secretes pepsinogen, which is inactive. Another part of the stomach epithelium secretes hydrochloric acid. Both of those are secreted into the interior of the stomach. The hydrochloric acid converts pepsinogen into pepsin, which is active, and now the pepsin can start digesting proteins.
One second. Okay.
Okay, so what happens when hydrochloric acid interacts with pepsinogen? The hydrochloric acid converts the pepsinogen into pepsin, which is active, and then that can start digesting proteins. Do you see what's the usefulness of this? Well, the point is the pepsinogen is created in the lining of the stomach, in the stomach epithelium, but we don't want it to start digesting the stomach epithelium. We don't want it to start digesting until it reaches the interior of the stomach. So the solution is that when the pepsin is first created, it's in an inactive form, pepsinogen. And it's not activated until it gets to the interior of the stomach. How does it know when it's in the interior? Because it meets up with the hydrochloric acid. And then the pepsin then, now that's activated, now can start digesting. But now because the pepsin is in the interior of the stomach, it's not going to do too much damage to the epithelium where it was created. Okay, does that make any sense?
Yes.
Okay, so so the key thing here is that the pepsinogen is created in a different part of the stomach's epithelium. It's created by different cells than the hydrochloric acid is. So while they're in the epithelium, they don't mix with each other because they're created by different cells. But they do meet when they get to the interior, and then the pepsin starts digesting. Okay, good. So here would be a good test question. What would go wrong if the epithelium secreted active pepsin rather than inactive pepsinogen? Why would it be bad for the stomach epithelial cells to create pepsin in its active form?
Then when hydrochloric acid wouldn't be secreted.
I don't think that. I think that's missing the point. So let's talk about that a little bit more. The problem, the whole point of this is if the pepsin was created in an active form in the epithelial cells, then they would digest themselves.
Okay.
Remember that pepsin is a highly digestive enzyme. It breaks things down. It breaks down proteins. Well, we want the pepsin to break down the protein in your food, but we don't want the pepsin to break down the proteins in the stomach's epithelial cells. Remember we were talking about how if the lining of the stomach does get broken down, that creates an ulcer. So the problem here is that the stomach epithelial cells are creating this very dangerous chemical. The stomach epithelial cells are creating pepsin, which is which can break down proteins. We want the pepsin to break down the protein in your food, but the danger is that it could break down the protein in the cell that just created the pepsin. So the solution to that is that when the pepsin is created in the stomach cells, in the epithelial cells, it's actually created in an inactive form, in the form of pepsinogen. And it doesn't get activated until it reaches the interior of the stomach. Now, the thing that activates it is the hydrochloric acid. So the key thing, maybe I should have made a note here, that different cells in the stomach epithelium secrete hydrochloric acid. The cells that create the pepsinogen are different from the cells that secrete the hydrochloric acid. So when the pepsinogen is created, it's not in contact with the hydrochloric acid yet. The pepsinogen doesn't come into contact with the hydrochloric acid until it reaches the interior of the stomach. Then it's activated, but at that point it's safe to be activated because now it's kind of outside the body. It's in the interior of the stomach, not in the epithelial cells themselves anymore.
Mm-hmm.
Okay, so let's see if we're understanding that. What would go wrong if the epithelial cells created active pepsin rather than inactive pepsinogen?
If they created active pepsin, it would start to break down. It would start to basically digest itself, which would cause ulcers.
Yeah, the epithelial cells would digest themselves, basically. If the epithelial cells were directly creating pepsin, the pepsin would immediately start digesting the cell it was created in, and that would be breaking down your own body cells. So instead, it's the epithelial cells secrete pepsin in an inactive form as pepsinogen, and then it's not activated until it gets to the interior of the stomach. What is it that activates the pepsinogen?
The hydrochloric acid.
That's right. So what's the difference between pepsinogen and pepsin?
Pepsinogen is the inactive form of pepsin.
That's right. And what activates the pepsinogen?
The hydrochloric acid.
That's right. Where is the pepsinogen created?
Stomach's epithelium.
Yeah, that's right. But it doesn't get activated until it reaches
the interior of the stomach.
That's right. By the way, there's a name that biologists use for the interior of a hollow organ. The interior of a hollow organ is called the lumen. So that's probably a term we should be using instead.
Mm-hmm. We talked about the lumen in class.
Good. So lumen just means the interior of a hollow organ. So what do we call the interior of the stomach? The stomach's lumen. Mm-hmm. And what do we call the interior of the small intestine? The small intestine's lumen. Okay. So this is what happens in the interior, also called the lumen of the stomach. Okay, good. This is a little bit more complicated than I made out. After the pepsin has become activated, the pepsin can now activate other pepsinogen molecules. The pepsin now is active. So what can this pepsin now do now that it's active? The pepsin, which is now active, now the pepsin can digest protein or convert other pepsinogen molecules into pepsin. So some of the activated pepsin will digest protein. Other pepsins will convert other pepsinogens into pepsin. So this is what's called positive feedback. That might be another topic from chapter 40. So this is a positive feedback loop because the creation of pepsin creates even more pepsin. The creation of the pepsin creates even more pepsin because pepsin can help to convert pepsinogen into more pepsin. So it's positive feedback because the more pepsin we have, the more that aids the creation of further molecules of pepsin.
1 second, my pen just ran out of ink.
Take your time.
Okay.
Okay. So, those are some of the key things that are happening in the stomach. So, what are the four main stages? What are the four stages of food processing?
Ingestion.
Good.
Oh, I thought you were writing it for some reason. Ingestion, digestion, absorption, and elimination.
Good. So obviously, ingestion begins in the mouth. Mm-hmm. What type of molecules are digested in the mouth?
Carbohydrates.
Yeah. By what enzyme?
Salivary amylase.
That's right. And salivary amylase is secreted by?
Salivary gland.
There you go. All right, and then the food passes out of the mouth into
The esophagus,
great. What do you call the chamber between the mouth and the esophagus?
the pharynx.
That's right. So it passes from the mouth to the throat into the esophagus. It's moved down the esophagus by what?
By the smooth muscle contractions.
Yeah, that's right. And what's the name for those smooth muscle contractions?
Um, peristalsis.
Peristalsis. That's right. Good. Okay, and then where does it enter after the esophagus?
The stomach.
Good. Okay. So what happens in the stomach?
The stomach will digest proteins by pepsin.
That's right. Where does the pepsin come from?
Oh, well, I'm forgetting it from. No, not from the. One second.
Take your time.
Oh, the stomach's epithelium.
Yeah, from the lining of the stomach epithelium. Good. What is it that prevents the pepsin from just digesting the epithelial cells where it's created?
The pepsinogen being inactive.
Yeah, so it's actually secreted in inactive form as pepsinogen. Good. And then what activates it when it gets to the interior?
The hydrochloric acid.
Yeah, where did the hydrochloric acid come from?
By the lining of the stomach.
From other cells in the stomach lining. Yeah. So it came from other cells in the stomach epithelium. Good. So what does the hydrochloric acid do to pepsinogen?
It activates it into pepsin.
That's right. And then remember that pepsin can activate further molecules of pepsinogen in a positive feedback loop. Okay. So what's the pH of the stomach?
2.
Yeah. Is that acidic or basic?
Acidic.
Yeah. What keeps it acidic?
What keeps it acidic? The hydrochloric acid.
That's right. So I guess the hydrochloric acid has that role as well. So what pH do you suspect that pepsin operates well at? Most enzymes operate well at a pH of 7, but what pH does pepsin operate well at?
2.
Yeah, because that's the environment of the stomach. So unlike most enzymes, pepsin operates well at a very low pH. What do we call the food matter when it's in the stomach?
Chyme.
Right, or acidic chyme, because it's very acidic at that point. Okay, very good. All right, so then what happens, what's the next stop after the stomach?
The small intestine.
Yeah, the small intestine. Good. Okay, good. And what types of molecules are digested in the small intestine?
Carbs, fats, and proteins.
Pretty much all of them. That's right. Good. Now we have to give some more detail now about the small intestine. The small intestine actually has 3 portions. There's the duodenum, the jejunum, and the ileum. Let's write this down somewhere. I guess I'll put that in this picture here. All of this digestion we've been talking about actually occurs in the first part of the small intestine, the duodenum. And then the rest of the small intestine is called the jejunum and the ileum. So there's 3 portions of the small intestine: the duodenum, jejunum, and the ileum.
Where does it say duodenum?
Farm today.
Oh, there it is. Okay. Let me. What is that? I can't. Is it No, I can't.
Jejunum. Jejunum. All of this type of digestion we were talking about occurs in the duodenum. And then most of the absorption occurs in the jejunum and the ileum. So this is some more detail that we hadn't gotten into before. So the pancreatic amylase is secreted into the duodenum. The lipase is secreted into the duodenum. The bile is secreted into the duodenum. Digestion of proteins continues in the duodenum. Digestion of fat continues in the duodenum. Digestion of proteins continues in the duodenum. And then digestion is basically finished in the duodenum. And then the function of the next two portions, the jejunum and the ileum, is absorption. Absorption of what? Well, absorption of all those monomers and water.
Would the ileum or the duodenum, would they have specific roles? Like, would they absorb into the lymphatic system and one absorbs into the bloodstream?
They both can absorb. I believe that they both absorb into both.
Okay.
So I believe that that's more detailed than your textbook goes into and more detailed than I know. But as far as I know, carbohydrates and proteins would be absorbed out of both the jejunum and the ileum into the bloodstream. Okay. And fat would be absorbed out of both the jejunum and the ileum into the lymphatic system. So we're not going to have to distinguish between these. You don't need to know what the differences are between the jejunum and the ileum. All you need to know is that they're the second and third portions of the small intestine, and their main function is absorption. So the main function of the duodenum is digestion, and then the main function of the small intestine and jejunum, of the jejunum and ileum, is absorption of monomers and water and other nutrients. Okay, you got that down so far?
Yes.
Okay, so where is pancreatic amylase secreted into?
Into the duodenum.
That's right. Well, what are the names of the three parts of the small intestine?
The duodenum, the jejunum.
Yeah, good.
And the ileum.
Yeah. What order does the food go through them in?
The duodenum, the jejunum, and the ileum.
Same as you just said. Okay, good. Where is the bile secreted into?
In the duodenum.
That's right. And where's the lipase secreted into?
In the duodenum.
That's right. Where's the pancreatic amylase secreted into?
In the duodenum.
Yeah, so all of these digestive enzymes are secreted into the duodenum because that's where the digestion is happening. And then the other portions of the small intestine are for the simpler process of absorption. Okay, good. Okay, very good. So maybe we should talk, so let's talk about what happens with absorption now. So now that all of these monomers have been digested, they need to be absorbed into the bloodstream or into the lymphatic system. So would you agree that it's important for absorption to, that the surface of the small intestine here has to have a very big surface area? Because absorption can only occur across the surface. Absorption here can only occur across the surface. So this portion of the small intestine especially has to have a very large surface area. So let's talk about that a little bit. So I think we've covered now the most important parts of digestion. So now we're going to talk in more detail about absorption, which we hadn't talked about before. Or let's see, actually before we do that, we'll come back to that in a moment actually. But let's see where we are here. So looking at our objectives here, how would you answer number 4?
Define and compare the 4 main stages of... Okay, so define and compare the 4 main stages. So in ingestion, you're basically taking in the food molecules, and then digestion begins. It starts in the mouth, the mouth will digest carbohydrates, and then it'll move down the pharynx and then through the down the esophagus and into the stomach, and digestion will continue to take place until
Digestion is continuing to take place where?
In the stomach and the
Good.
small intestine.
Good.
And then at the end of digestion, the small
What does
intestine.
digestion mean? So I think here they want a little bit less detail. What does digestion mean?
Breaking down a food molecule.
Good. Into what?
Into sugar.
Or more, that would just be for one type. So into
Yeah.
monomers might be a good thing to put in your answer there. We're taking the polymers in the food and breaking them into monomers.
Okay.
So what does digestion mean? Digestion means basically breaking polymers into monomers.
Okay.
Okay, good. And then what's the third stage after digestion?
Absorption, and that's when the broken down when the monomers are either absorbed into the bloodstream or the lymphatic system.
That's right. Good. Now, this part here, you might be focusing on that business about the lymphatic system and the blood system. That's specifically for humans or other mammals. And here, they might want you to have a more general description that applies to any animal. So we could just say that, but even so, that's still a good answer. So the point is, absorption is when the monomers are absorbed out of the digestive tract and into the body cells. Different organisms would do that in different ways.
Okay.
And then what's the last stage after absorption?
Elimination. And
Right.
That's basically when the food that hasn't necessarily been broken down is eliminated from the body.
That's good. So the key thing is you said we eliminate the parts of the food that hasn't been digested, because or the parts that haven't been absorbed. The parts that haven't been digested could not be absorbed. Okay, good. All right. So here's another objective. Describe the common processes and structural components of the mammalian digestive system. That's kind of a vague idea, but that's kind of what we've been doing all this time. We've been talking about, so the common components are things like the mouth, the stomach, the small intestine, and we've been talking about what their roles are. So I think we've done a pretty good job with that. Here's number 8. Compare where and how the major types of macromolecules are digested and absorbed within the mammalian digestive system. So we spend a lot of time on that. So what could you say about that?
Compare when and how the major types of macromolecules are digested and absorbed. Okay.
Notice how this objective here is specifically about mammals, whereas this one up here was just in general. So here we were just talking about ingestion and digestion absorption for any organism, not just mammals. But now we're talking more about the specifics of what we've been talking about in most of the tutoring. So how could you start answering this? This would be a good essay question for a test.
It would begin in the mouth, where specifically carbohydrates would be digested.
Good.
So once carbohydrates are
By
digested,
what key enzyme?
Salivary amylase
From the...
from the salivary glands.
Okay, good.
So compare where and how. So the carbohydrates will be broken down in the mouth, will be digested in the mouth by the salivary amylase, and then the carbohydrates as well as other food molecules will travel down the esophagus into the stomach, where proteins, if proteins are there, will be broken down by the pancreatic. No, that's carbohydrates. So proteins are broken down in the stomach by pepsin.
Good. That's important to remember. Good.
And hydrochloric acid is secreted by the lining of the stomach, which activates pepsin and helps break down the proteins.
Good.
So digestion continues moving down into the small intestine, where all the main types of macromolecules will be digested in the small intestine. For carbs, the pancreatic amylase is what breaks down the carbohydrates. The fats are broken down by bile salts. They're emulsified. They turn the big globs of fat into small droplets covered with the bile salts. The lipase will then break down those. The pancreatic lipase will then further break down the fat droplets, turning into glycerol and fatty acids. It absorbs out of the small intestine into the lymphatic system.
Great. Good.
Um,
Good. So you
yeah.
just mentioned that the fats were absorbed into the lymphatic system, and carbohydrates and proteins are absorbed into what?
The bloodstream.
That's right. Out of where?
Out of the small intestine into the bloodstream.
That's right. Good. Okay, so those would be some of the key things that you'd want to say here. So proteins are ultimately digested into what?
Proteins are ultimately digested into amino acids.
That's right. Those are the monomers of proteins.
Mm-hmm.
And carbohydrates are digested into what?
Monosaccharides.
Those are the monomers of carbohydrates. Good. And fats are digested into?
Glycerol.
And
3 fatty acids.
That's right. Those are the components of a fat molecule. Okay, good. Let's see. We talked about this a little bit last time. What are the major functions of the large intestine?
Oh, the major function of the large intestine is basically the absorption of water.
Okay, yeah, that's right. All right.
And I guess processing undigested food out of the rectum and out of the anus.
So it takes the undigested food and it creates that, by absorbing the water, it's forming the feces. That's right.
Mm-hmm.
Okay, so here's something we were talking about. So here's kind of one of the key diagrams we've been talking about. Food goes from the mouth to the esophagus, to the stomach, to the small intestine, to the large intestine, through the rectum, through the anus. The key glands here, the salivary gland delivers into the mouth. The liver delivers into the gallbladder, which then delivers into the small intestine. The pancreas delivers into the small intestine. Between the mouth and the esophagus is the pharynx. And obviously this is just a cartoon. It doesn't show the actual shapes of the organs. Here's what the small intestine actually looks like and what the large intestine actually looks like. What's the first portion of the small intestine that the food enters after the stomach?
Of the sphincter?
Now, this is something I'm asking you from memory. What if we said, remember the small intestine has 3 parts. What's the first part of the small intestine that the food enters after the stomach?
The duodenum.
Right. So notice the sphincter is a muscle that controls the passage between the stomach and the duodenum. We haven't talked about that much. But there's a muscle here that closes the entrance. So while the food is being digested in the stomach, the sphincter would be closed. And then when some of the food is ready to pass into the small intestine, the sphincter muscle would relax somewhat and let the food travel from the stomach into the duodenum of the small intestine. So that's what they have labeled here. So here's the stomach. What is secreted into the interior of the stomach?
The hydrochloric acid, or the pepsin.
Yeah, but the pepsin is not secreted as pepsin. What form is it secreted as?
Pepsinogen.
So notice this is a picture of the epithelial lining of the stomach. And you can see these red cells here are secreting pepsinogen into the lumen. And then this yellowish cell here, that's secreting the hydrochloric acid into the interior. And remember, what did we say? What does this little diagram show happening?
The pepsinogen converting into pepsin.
Yeah, what's converting it?
HCl.
That's right. And what do you think this arrow represents?
Pepsinogen.
Remember that the pepsin can activate more pepsinogens.
Oh yeah.
It's not a very clear arrow, is it?
Yeah.
So remember, what converts, so to speak, the first molecule of pepsinogen into pepsin?
Hydrochloric acid.
But then, what could this pepsin do?
Activate more pepsinogen.
and create more pepsin. This is the positive feedback loop that we were talking about before. So this is a better drawing of what, here's the lumen or the interior of the stomach. And notice that the lining has these big infoldings lined with, here's the epithelial cells. Here's a mucus cell. We talked about how there have to be cells that constantly create mucus that protect the lining. And then we have these chief cells here. And then we have other cells, chief cells, that are creating the pepsinogen, and parietal cells that are creating the hydrochloric acid. And those are all secreted into the interior of the stomach here. Okay, so that's an idea that we were talking about before. Here's the liver. How does the liver aid in digestion?
The liver will secrete bile.
And where does the bile go?
The gallbladder.
So here's a picture that's more realistic of their relation in space. The bile would first go here and then up into the gallbladder, and then what?
The gallbladder will send over the bile into the small intestine.
Good. Into which portion of the small intestine?
The duodenum.
Right. The first portion of the small intestine is the duodenum. What do we call the food matter as it's passing through the stomach?
Chyme.
Here's the acid chyme. And who else is secreting into the duodenum besides the gallbladder?
The pancreatic.
So here's the pancreas that's also secreting into that same place. What are some of the things that the pancreas secretes?
Pancreatic amylase.
Right. That digests what type of molecule?
I'm sorry, could you repeat that?
What type of molecule gets digested by pancreatic amylase?
What molecules
Yeah.
I'm sorry.
Proteins, lipids,
okay.
Carbohydrates. What type of molecule is digested by pancreatic amylase?
Carbohydrates.
That's right. Good. Anything else secreted by the pancreas that we talked about besides pancreatic amylase?
What else is secreted? Yes. The lipase.
Also lipase, and that digests?
The lipids.
Right. And there's actually lots and lots of other stuff, enzymes, that we didn't get into that it also secretes. But those are some of the key ones that are likely to be tested here. Okay. Notice something interesting here. The small intestine here is 6 meters in length. I guess that's about 18 feet. So if you think about how tall a 6-foot tall person is, the small intestine, if it was stretched out, would be as long as 3 6-foot tall people. How does it fit in the body? It fits in the body because it's extensively wrapped around itself and curved and folded like this. It has to be long to give it a long enough space for all the absorption that we want to occur there. Okay, so what's digested in the mouth?
Carbohydrates.
Yeah, by what type of enzyme?
Salivary amylase.
Yeah, what do we call a carbohydrate polypeptide? Oh, sorry. What do we call a carbohydrate polymer?
Polysaccharide.
Right. And what are some of the most important polysaccharides that get digested? We talked about 2 main types of polysaccharides.
2 main types of polysaccharides?
Right. We talked about starch and glycogen.
Oh, glycogen. Yeah.
Remember that starch is how plants store energy, and glycogen is how animals store energy. All right, so this is an important table that summarizes some of the stuff that we've been talking about. So in the mouth, carbohydrate digestion occurs of these key polysaccharides, starch and glycogen, and it cuts them down into smaller polysaccharides. Notice that, is there any digestion of proteins or fats in the mouth?
No.
So these are blank. Okay, good. And then is anything digested in the stomach?
Proteins.
Yeah, who digests the proteins?
The pepsin.
Right. So here they have proteins in the stomach digested by pepsin into smaller polypeptides. And is any other types of molecules digested in the stomach besides proteins?
No.
No, not to any large extent. That's why there's blanks in these positions. Okay, and then what types of molecules are digested in the small intestine?
All the ones we talk about?
Yeah, pretty much all of them. Good. So polysaccharides are digested by what type of enzyme?
Carbs by the pancreatic amylase.
So the polysaccharides here are digested by pancreatic amylase, which comes from what gland?
The pancreas.
That's right. Good. And proteins are also digested in the small intestine. I didn't talk about these particular names with you. I don't know whether these would be tested or not, so I don't want to go into too much detail. I'm not sure whether you really need this much detail for the test or not, but it's here in this table. I think you said your instructor said not to worry about the nucleic acid digestion much, but that also occurs in the small intestine. Mm-hmm. And then how are fats digested in the small intestine? What happens first? What's the process of fat digestion?
During fat digestion, there'll be like a giant glob of fat,
right.
And in order for that to be broken down, the bile salts will kind of coat the outer layer of the fat, because they can't have access to the inside of the fat, so they coat the outside, and that allows the fat to be broken down into little tiny droplets of fat. And then the lipase, which comes from the pancreas,
good.
It digests the fat droplets, and then those fat droplets become glycerol and fatty acids.
Great.
Those are absorbed out of the small intestine and into the lymphatic system.
Excellent. Good. So notice that that's all summarized here. This is what we talked about. The fat globules are emulsified into droplets by the bile salts. And then the lipase breaks down the fat droplets into glycerol and fatty acids. Good. So what is the source of the digestive enzymes in the small intestine? Where do the digestive enzymes in the small intestine come from where?
I'm sorry, could you repeat that?
The digestive enzymes in the small intestine come from where? Where are they created or secreted from?
The small intestine are secreted from the epithelium?
Partly from the epithelium, that's right. Some of the enzymes come from the epithelium. Some enzymes are coming from the epithelium of the small intestine, and the other ones are coming from
the pancreas?
That's right. So notice here they have a specific category for some enzymes that are attached to the epithelium of the small intestine. So you can see this table goes into even more detail than we went into, but I went into the details that I think are most important for the exam. Basically, hopefully these extra details you don't need to have memorized, but a lot of the stuff on here we talked about together. So carbohydrates are ultimately digested into what?
Into glucose or sugar.
What's the name of a carbohydrate monomer?
Monosaccharides.
That's right. So what are carbohydrates digested into?
Monosaccharides.
That's right. And what are proteins digested into?
Proteins are digested into amino acids.
That's right. And fat is digested into?
Fatty acids and glycerol.
So notice at the end of the arrows here, at the bottom of the carbohydrate column, we end up with monosaccharides, just like we were saying. At the bottom of the digestion column, we end up with amino acids, just like we were saying. At the bottom of the fat column, we end up with glycerol and fatty acids, just like we were saying. So this table summarizes a lot of the key things we were talking about, although it maybe has maybe it has more detail than you actually need on the test. I don't know. It's hard for me to tell exactly how much detail your instructor wants. Okay, good. So those are the aspects of this that we've talked about so far. So there's lots of details here. There's a bunch of stuff we haven't talked about here yet, but I've been trying to focus on the stuff that I thought was most important so far. Okay, good. So that would be important stuff for you to go on and review. The key thing here is I think we made good progress today, but it's hard to remember it all. So it would be important to go back and review all of these key ideas that we talked about here. I guess these are going to be pretty important on the next test. So in the meantime, so I think you'd already done a pretty good job of reviewing what we went over last time. You were remembering most of what we talked about for digestion, but obviously we covered a bunch of new stuff here today. So as usual, the first thing we'll do in the next session is quiz ourselves on this material to make sure we still have it down. So we went over a lot of new stuff today, so it would be good to keep going back over all that material. I think the best way to memorize this is to try writing it out on paper. So try writing out this sequence here that we have for, this organizes a lot of the work that we have for how things go through the digestive tract. Also, if you take a look at your lecture notes for that chapter now, for chapter 41, excuse me, hopefully some of the lecture notes will now make more sense now that we kind of have a background, so you might be able to memorize more stuff. And let's see, what else is the key stuff that we went over here. We talked about the activation of pepsin in the stomach. That's pretty important. That's very likely to be tested. We talked about digestion of fats, first emulsification by bile salts, and then digestion by lipase. That's pretty important. That's likely to be tested. So make sure to try to review this as carefully as you can. What is one of the types of large biological molecules?
Carbohydrates.
What's another name for a carbohydrate polymer?
Polysaccharide.
And what's another name for a carbohydrate? What's the name of a carbohydrate monomer?
Monosaccharide.
Very good. What's another type of large biological molecule?
Lipids.
Okay, excellent. That's right. What's the main type of lipid that we focus on in the digestion chapter?
Fats.
Okay, good. What's another type of large biological molecule?
Proteins.
What's another name for a protein polymer?
Polypeptide.
And what's the name of a protein monomer?
Amino acid.
Okay, very good. What's the last type of large biological molecule?
Nucleic.
And what are the two types of nucleic acid polymers?
DNA and RNA.
Excellent. And what's the name of a nucleic acid monomer?
Hello?
I'm here. Can you hear me? I was asking, what's the name of a nucleic acid monomer?
Oh, nucleotides.
Okay, excellent. Good. So those are just important ideas to keep reviewing because they're so crucial for the course. It looks like we're still quite comfortable with that. That's good. What are the four main stages of food processing?
Ingestion. Digestion. Absorption, and then elimination.
What do those four terms mean?
Ingesting is basically taking the food and consuming it, so it's in your body. Digestion is the breakdown of the food. Absorption is...
Let's be a little bit more specific of what we mean by breakdown. So in most cases, what that means is breaking down the polymers into monomers. That would be a good point to make there. Okay, good.
And then absorption would be basically everything that was digested. So the monomers breaking down to polymers. They would basically be absorbed into the bloodstream or the lymphatic system.
Out of the digestive tract. Good. So absorption is when the monomers... So digestion occurs... Where does digestion occur? So what is ingestion? Ingestion is when the food is inserted into the digestive tract.
Mm-hmm.
Then digestion, where does digestion occur? Digestion occurs in the digestive tract, also called the alimentary canal. And digestion is when the polymers are broken down into monomers. Then absorption is when the monomers are absorbed out of the digestive tract and into the body, into the bloodstream or the lymphatic system. Okay, good.
And then elimination is when the foods that cannot be broken down are eliminated through the body or eliminated from the body through the anus.
Very good. That's right. Now, these stages occur for basically all animals, not just not just mammals. So in most large animals, they would be eliminated through the anus. In very small animals, they might be eliminated in some other way, like a single-celled organism. They would be eliminated in some other way. But these are the 4 basic stages that are happening in the digestive process. Okay, very good. So what's eliminated here is the food matter that could not be broken down or absorbed. Okay, good. Okay, so what types of biological molecules are digested in the mouth?
Carbs.
Good. So what molecule assists in the digestion of those carbohydrates?
Salivary amylase.
Excellent. What type of molecule is salivary amylase?
An enzyme.
Excellent. And what type of molecule are enzymes?
Enzymes are proteins.
Excellent. Great. Good answers. What gland secretes the salivary amylase?
Salivary gland.
Okay, excellent. Okay, so then where does the food go after the mouth?
Then it goes down the pharynx, through the esophagus, and into the stomach.
Good. How is the food moved through the esophagus?
Through smooth muscle contractions.
Excellent. And I think we'll need to know the technical name for those smooth muscle contractions. Do you know what the name of those contractions are?
Yeah, 1 second. Hold on. Do you remember talking about it? First, peristalsis.
Peristalsis. In fact, peristalsis is what moves the food through the entire digestive tract, not just the esophagus. Okay, so that's a term that we'll need to have memorized. That could certainly be tested. So to review, what's the name of the process that moves the food through the digestive tract?
Peristalsis.
That's right. Okay, good. So then the food ends up in the stomach. So what type of molecules are digested in the stomach?
Proteins.
And what molecule helps to digest those proteins?
Hydrochloric acid and pepsin.
Excellent. Good. Good. What type of molecule is pepsin?
An enzyme.
Excellent. And what type of molecule are enzymes?
Proteins.
Excellent. Very good. Okay. So this is something we worked on last time. So what does pepsin do?
Pepsin allows the proteins to be broken down in the stomach.
Yeah, it starts breaking the proteins down into smaller chains.
Mm-hmm.
Now, where did the pepsin come from?
Pepsinogen.
That's right. But where was it secreted from?
The epithelial, the linings of the stomach.
The epithelial lining of the stomach. That's right. The pepsin. So we talked last time about how the epithelial tissues are tissues that line organs or cavities. So around the exterior of the stomach, there's a layer of epithelial cells. Those epithelial cells secrete pepsin. And where does the hydrochloric acid come from?
Also comes from the epithelial of the stomach, the lining.
That's right. It also comes from the epithelium of the stomach. So if the pepsin is coming from the epithelium of the stomach, what prevents the pepsin from digesting the lining of the stomach?
No.
I'm sorry, could you repeat that?
Remember that pepsin digests proteins, and the pepsin was created in the epithelial tissue of the stomach. So what prevents the pepsin from just digesting the stomach's own proteins and eating a hole in the stomach?
Because pepsinogen is inactive, that's what prevents it. So before the activation of pepsin, before the HCl is released, pepsin is only pepsinogen, so the acidity isn't. It won't break down the stomach before it actually is released into the into like the inner. Could you say the lumen?
That's right. You could say the interior of the stomach, but we discussed last time that the technical name for the interior of an organ is the lumen. That's right. Yeah. Okay, good. So the stomach epithelial cells, as you were saying earlier, don't actually secrete pepsin. They secrete pepsinogen, which is a longer version. And is pepsinogen active or inactive?
Inactive.
It's inactive. That's right. And then what activates the pepsinogen?
The hydrochloric acid being released.
Right. So let's make sure we're clear about this. Who secreted the hydrochloric acid?
The epithelial of the stomach.
Good.
The epithelial lining.
Epithelial cells in the lining. Good. One thing maybe I didn't quite emphasize before is different cells then secrete the pepsinogen. Oh, yeah. Otherwise, the whole system wouldn't work. If the hydrochloric acid was being made in the same cells that were making the pepsinogen, then the hydrochloric acid would get to the pepsinogen too soon while it was still in the epithelium. So there's one set of cells that creates the pepsinogen and another set of cells that creates the hydrochloric acid. If I remember correctly, these cells are called chief cells, and these cells are called the parietal cells. I haven't been writing that down because I don't want to put it down too much detail. I'm not sure if you'd be tested on that detail or not. But anyway, you should know that these are two different types of cells. And then they're both secreted into the lumen. And in the lumen, what happens is that the HCl, the pepsinogen, plus hydrochloric acid come together. The hydrochloric acid breaks off a piece of the pepsinogen and turns it into pepsin. And I'll put an asterisk here to show that the pepsin is the activated form. So what creates pepsin? Pepsin is created when hydrochloric acid reacts with pepsinogen. Now, do you remember there was one other step here that was also creating more pepsin? What was the other thing we discussed that was creating more pepsin besides the hydrochloric acid reacting with pepsinogen?
The pepsin itself.
Yeah. What does the pepsin do? The pepsin can now react with another molecule of pepsinogen. And that turns that new molecule of pepsinogen also into pepsin. So those would be important steps to include in an answer if they were asking you to trace out this process. So would this be called a positive feedback loop or a negative feedback loop?
Positive feedback loop?
Positive, because the more pepsin there is, the easier it becomes to make even more pepsin. Making more, so an increase in pepsin makes it easier to increase the amount of pepsin even further. The advantage of that is that if any proteins have entered into the stomach, we want to activate a bunch of pepsin really fast to start digesting it, because any stuff that doesn't get digested is not going to be able to be absorbed. Okay? Okay, good. So those are all pretty likely to be tested. That's all review. Now let's try to add a few more details that we didn't get to last time. One of the other objectives we haven't gotten to yet is hormonal control, but that's probably going to be important on the exam. So how does the stomach, how do these cells, how do the chief cells and the parietal cells know to secrete pepsinogen and hydrochloric acid? Well, they're receiving a hormone. There's a hormone that stimulates them to secrete these gastric juices. Do you happen to have learned in your class yet what the hormone is that stimulates the stomach?
No, I don't think so. No, we didn't go over that. That's no.
Okay, or you might just not remember it. You and I haven't talked about it, but that was one of the objectives. So let's talk about that. So what happens is, do you remember, what do we call the food matter when it enters the stomach?
Chyme.
Chyme. That's right. So Although maybe it's especially called that after it mixes with the acid. But anyway, we'll say here that the presence of food in the stomach causes the stomach epithelium to secrete gastrin. So we're secreting here gastrin. Gastrin is a hormone. Gastrin is a hormone. And then the gastrin stimulates these two parts. The gastrin stimulates the chief cells to secrete pepsinogen and the parietal cells to secrete hydrochloric acid. So I think there's a good chance that would be tested, this hormonal control idea. Does that make sense?
Mm-hmm.
Okay. All right. So how do these cells know when to secrete pepsinogen and hydrochloric acid?
So with the presence of food in the stomach, it'll cause them. It'll activate them because they'll know that there's food in the stomach and they need to release the HCl and the pepsinogen in order to break down the food.
That's good, but there's some missing steps there that I think they'd want on the test. So the key thing you left out was the gastrin. We need to describe its role. So when there's food in the stomach, that causes the stomach to secrete gastrin. And then the gastrin stimulates the stomach to secrete pepsinogen and hydrochloric acid. So the food leads to the secretion of gastrin. The gastrin leads to the secretion of pepsinogen and hydrochloric acid. So what type of molecule is gastrin?
It's a hormone?
It's a hormone. We haven't talked much about hormones before, but a hormone is a messenger. Well, I guess we did talk about hormones for plants, right? We talked about plant hormones. We talked about how they bring messages and signals in plants. Well, obviously hormones play an important role in animal biology as well. So gastrin is a hormone that sends this message. All right, so what is the hormone that controls the release of the gastric juices from the stomach?
Gastrin.
Gastrin, that's right. Okay. All right, so then moving on, what's the next element? Where does the food go after the stomach?
The small intestine.
Excellent. However, last time we saw that it's important to break up the small intestine into 3 segments. What do we call the first segment of the small intestine?
The duodenum.
Right.
What? Oh, duodenum.
Okay, very good. So let's write that down. Okay. I don't know who's correcting you there, but the glossary says it's duodenum. So I think your pronunciation was correct. Maybe they're both acceptable. But your textbook's glossary pronounces it duodenum. Okay, so
It was my brother.
Okay. Well, that he might be using an alternative pronunciation. Okay, so let's see here. Here we have duodenum. So this is the first part of the small intestine. Okay, so what's going to happen in the duodenum?
In the duodenum, the food, whatever food wasn't digested completely in the stomach, will be digested. Is where basically the rest of digestion will take place in the small intestine.
Good. So let's be more specific. What's one type of molecule that's digested in the duodenum?
Carbs.
And what would be one type of molecule that aids in the digestion of those carbohydrates?
Oh, hold on. Pancreatic amylase?
Yeah, that's an important molecule. What type of molecule is pancreatic amylase?
An enzyme?
It's an enzyme. Very good. Excellent. Who secretes the pancreatic amylase?
The pancreas.
That's right. So the pancreatic amylase was secreted by the pancreas. Good. Okay. Okay, fair enough. What's another type of molecule that is digested in the small intestine?
Proteins.
And again, this is actually all pretty much happening in the duodenum.
Mm-hmm.
That's really the key element here, digestion of protein. And your textbook lists a bunch of specific enzymes that help to digest proteins here. I don't know if you'd be tested on those or not, but I'm not going to write those down. Okay, digestion of proteins. Good. And what's another type of molecule that's digested in the duodenum?
Fats.
Okay, that's right. Now remember, the digestion of fat is a little bit harder or different from other types of digestion. So let's go into that in a little bit more detail. Let's outline what's the process of the digestion of fats in the duodenum.
During the digestion of fat, there would be one large fat globule. And in order for that to be broken down and digested, the liver will create bile, and then the bile that is stored in the gallbladder will be released, and then the bile that is stored becomes the bile salts.
It includes bile salts. Good.
Includes bile salts, and these bile salts will coat themselves around the large globule, and then they'll break down that large globule into smaller like fat droplets.
Or smaller globules. Good.
Yeah. And then the pancreas will secrete like lipase, and then that lipase will allow it will digest the fat droplets even more to create glycerol and 3 fatty acids.
Excellent. What type of molecule is lipase?
An enzyme?
That's right. What does it do?
Sorry, could you repeat that?
What does lipase do? What does lipase do?
Fat molecule.
Good. Why is lipase a logical name for lipase?
Lipids.
Yeah, fats are lipids. So I just wanted to point out this is not too hard to remember that it digests fats because those are a type of lipid. That's the reminder in that name. Okay. Excellent. Okay, yeah, so that's those, the breakdown of those into glycerol and fatty acids. Remember that, okay, good. So those are some key steps. Now, and so those are delivered by, I think you already explained how the bile was produced in the liver, and then the bile is stored in the gallbladder. Okay, good. I suppose there's also digestion of nucleic acids here as well, but I think your course isn't really focusing on that, so we'll move on. Now, what else can we say here? So remember, what was the pH of the stomach?
2.
Yeah, is that acidic or basic?
Acidic.
Yeah, much more acidic than normal biological conditions. What's kind of a more normal biological pH?
Would it be 4?
Actually, normal biological pH is pretty close to neutral. Do you remember what a neutral pH is?
7.
Yeah, so I think normal biological pH is like 7.2 or 7.4, not too far from neutral. Okay, so what pH does pepsin operate well at?
2.
2, because it's designed to be used in the stomach.
Yeah.
But what pH would you expect most enzymes to operate best at?
7.2.
Yeah, because that's what the normal pH is in most places. Okay, now, so the key thing here is that the small intestine does not remain acidic. It has to become basic in order for the enzymes here to do their work, like pancreatic amylase and lipase. So something has to release a base to neutralize the acid from the stomach. So what's going to happen here, I don't know, do you happen to remember what is it, what are the steps that neutralize the acid?
No, I don't believe you talked about it in class yet.
One of the other objectives here that we need to be familiar with, the pancreas now is going to release what's called bicarbonate. The formula for bicarbonate is this. And bicarbonate is a base. So when the acidic chyme moves out of the stomach and into the duodenum, it's going to carry along a lot of hydrochloric acid. So it's going to at first be highly acidic. But in order for the pancreatic enzymes to work properly, we have to bring the pH back closer to neutral. So the pancreas releases bicarbonate. Here's the formula for bicarbonate. I'm not sure why this is called bicarbonate, but anyway, that's the conventional name for it. This is a base that neutralizes the hydrochloric acid from the stomach. Okay, do you have that in your notes? Take your time.
1 second. Okay.
Okay. All right. So the pancreas releases this bicarbonate that neutralizes the acidic chyme and brings us closer to neutral. Okay. So where does the bicarbonate come from?
The bicarbonate comes from it's the HCO3. The duodenum.
Actually, it's being secreted into the duodenum, but it's coming from the pancreas. The bicarbonate is coming from the pancreas. And where is it secreted into?
It's released from the pancreas and secreted into the duodenum.
That's right. Good. And what's the purpose or function of the bicarbonate?
It's to neutralize the HCl from the stomach.
Good. So does it increase or decrease the pH in the small intestine?
Decrease. Oh wait, no. If it's neutralizing the HCl, then you're increasing it.
So let's review those ideas about pH. So what would be a neutral pH?
7.
So what would be an acidic pH?
2.
Or less than 7?
Yes.
And what's a basic pH?
Anything more than 7?
That's right. Obviously, the higher, if it's still 7.2 or 7.4, that's close to neutral. But the more it is above 7, the more basic it is. By the way, another name for basic is alkaline. In biology, oftentimes basic is called alkaline. Okay. So when you add acid, what does that do to the pH? Increase it or decrease it?
Decrease it.
And when you add base, what does that do to the pH?
It increases the pH.
Great. So when you add hydrochloric acid, what does that do to the pH?
It lowers it.
Good. And when you add bicarbonate, what does that do to the pH?
It neutralizes it.
Well, it'll neutralize what might be there, but what does it do if you start with the acidic chyme, the pH in the stomach was 2, and if you add bicarbonate to that, what's that going to do to the pH? Increase it or decrease it?
Increase?
Increase it. I'm not actually sure exactly how high it increases. I'm not sure if it increases all the way up to 7 or not, but in any case, it's going to increase the pH closer to neutral, or maybe even slightly basic. Okay, so what does the bicarbonate do to the pH in the duodenum?
It neutralizes the HCl from the stomach.
Good. And what does that do to the pH?
It increases it.
That's right. Okay, good. So what's the name of the substance that the pancreas releases to change the pH of the duodenum?
Bicarbonate.
Bicarbonate. Okay, good. All right, so those are all fairly important ideas that we didn't have a chance to get to last time. Very good. Okay, and now something else we have to discuss is, again, the hormonal control. So remember, what hormone controls the operations in the stomach?
Gastrin.
Gastrin, that's right. So now we need to go over what's the hormonal control of the duodenum. So there's a couple of different enzymes here. So let's see, the pancreas, no. So I think the epithelium of the duodenum, when the epithelium senses food matter entering into it, that's going to release two different enzymes, CCK and secretin. Okay, so what does the CCK do? The CCK goes to, maybe I should write it right here. So the CCK goes to the gallbladder, and it has a positive effect on the gallbladder. So what would it cause the gallbladder to do? What's the function of the gallbladder?
Store bile.
So what would the CCK cause the gallbladder to do? Release the bile, right?
Okay.
So when there's food in the small intestine, that causes the secretion of CCK. That goes to the gallbladder and causes the gallbladder to release the bile. The CCK also goes to the pancreas. It has a positive effect on the pancreas, and it causes the pancreas to release pancreatic enzymes.
Mm-hmm.
There's also another hormone that's also being released by the small intestine here called secretin. The secretin also goes to the pancreas. It also has a positive effect on the pancreas, but it causes the pancreas to release something else besides enzymes. Do you remember we were just talking about what's the other thing the pancreas releases besides enzymes?
Hormones?
Let's see. It does release hormones, but that's not what we're talking about right now. The other thing the pancreas releases for the digestive process here is bicarbonate. So there also has to be a signal to tell the pancreas to release the bicarbonate.
Mm-hmm.
And remember, this is the symbol for bicarbonate. So CCK and secretin are hormones. Tell me when you have that in your notes.
I have that.
So what's one of the targets of CCK?
The gallbladder.
That's right. Does CCK have a positive or negative effect on the gallbladder?
Positive.
And what does it cause the gallbladder to do?
To release the bile.
Good. Into the duodenum. And then what's another target of CCK?
Pancreas.
Good. And does it have a positive or negative effect on the pancreas?
Positive.
And what does it cause the pancreas to do?
Pancreas will release bicarbonate into the duodenum.
So that part looks like we haven't quite got memorized yet. Can you read my table here? What does this table say that the CCK is causing the pancreas to do?
Oh, it's a positive to release enzymes.
That's right, not the bicarbonate. What causes the pancreas to release bicarbonate?
The secretin.
Yeah, that's what I was trying to show with this diagram here. Okay. So what's one of the targets of CCK?
The gallbladder.
Good. What's another target of CCK?
The pancreas.
Good. What does the CCK cause the pancreas to do?
To release enzymes.
Good. And then what's one of the targets of secretin?
Pancreas.
Good. And what is the does secretin have a positive or negative impact on the pancreas?
Positive.
Good. And what does it cause the pancreas to do?
Release HCl3.
Yeah, otherwise known as bicarbonate, into the duodenum. Okay. When would the small intestine secrete CCK and secretin? It secretes them when when food matter, when the chyme is entering into the duodenum. You wouldn't want all of these enzymes in bile and bicarbonate to be released if there's no food in the duodenum, because then they'd just be wasted. So these hormones send the message that tell these glands when to secrete the various substances. Okay, good. Let's see, is there anything else they said about that? And there was one other thing. Okay, good. Also, though, now that there's food in the small intestine, we want to tell the stomach to slow down and not send any more food. We don't want to send too much food into the small intestine at once, or there won't be time to digest it and absorb it in the small intestine. So while there's food in the small intestine, we need to slow down digestion in the stomach so that it's not going to send even more food in this direction. So both the CCK and the secretin, they both also have targets in the stomach. Based on what I was saying, would the CCK and secretin have positive or negative effects on the stomach?
Positive.
Let's go through that logic again. When are CCK and secretin secreted?
When the food enters the duodenum.
That's right. So when there's food in the duodenum, where did that food come from?
The stomach.
Now that there's food in the duodenum, do we want to send even more food into the duodenum? Well, no. We need to, so the stomach, the entrance between the stomach and the duodenum is controlled by a sphincter muscle that can open or close to let more or less food out. We don't want to overload the duodenum, so to speak, with too much chyme, or it won't have time to digest the food matter that it has. So when there's food in the small intestine, do you want digestion in the stomach to increase or decrease?
Decrease.
You want it to decrease. You want the stomach to slow down to give the small intestine time to catch up, basically.
Mm-hmm.
So when there's food in the small intestine, do you want to have a positive or negative feedback back to the stomach?
Negative.
So should the CCK and the secretin have positive or negative effects on the stomach?
Negative. They should have a negative effect.
So here we have these two negative effects. Okay, I probably could have been neater here. I hope this diagram makes sense. So why don't you study this for a few seconds. Tell me when you feel that you have these ideas down, or if they're not making sense, I'll try to explain them more clearly. My diagram could have been neater.
I understand.
Okay. Okay, well, describe the hormonal control, the role of hormones in the duodenum.
The duodenum digestion of the carbohydrates. The duodenum will also digest proteins.
Good.
They can digest
That's good.
fat.
That wasn't quite what I was going for, though. I think you have those things down. So what I want to do now is review the material about hormones that we were just talking about. Maybe you haven't quite memorized that yet. So now would be a good time to memorize these ideas about the hormonal control. So where are the CCK and the secretin secreted from? They're secreted so these are secreted by the small intestine. And then what are their targets?
Their targets are for the CCK, the targets are the gallbladder and the pancreas. For the secretin, the target is the pancreas.
Good. And the CCK and secretin have one more target as well.
Stomach.
That's right. What does the CCK do to the gallbladder?
It allows the release of bile.
And what does it do to the pancreas?
That releases enzymes.
And the secretin?
Releases HCO3.
And how do they affect the stomach?
They affect the stomach because they tell the stomach when to stop digesting so the smaller intestine can catch up.
Yeah, they slow digestion in the stomach.
Mm-hmm.
Okay. So now I'll take that off the screen. So again, let's just go through that again just to make sure we have it memorized. Describe the hormonal control that's involved in the small intestine.
In the small intestine, there are the two hormones, CCK and secretin, and both of them have multiple targets. The CCK
when
targets.
Would CCK and secretin be secreted?
They'll be secreted when there is food matter inside the small intestine.
Good.
The CCK targets the gallbladder and allows the release of bile. The CCK also targets the pancreas. It'll cause the pancreas to release enzymes. The secretin targets the pancreas also. It's positive and it allows the release of bicarbonate from the pancreas. The CCK and secretin also have a negative effect on the stomach. The CCK and the secretin both stop the stomach from digesting.
Why would you want them to do that?
So the small intestine could catch up.
That's right. So that you don't want even more food to be exiting the stomach into the small intestine when the small intestine just got a delivery of chyme to work on. Okay, good. And then, so what type of molecule is CCK?
A hormone?
Good. And what type of molecule is secretin?
A hormone.
That's right. And what was the hormone that was regulating this? What was the hormone that was secreted by the stomach to regulate the stomach?
Gastrin?
That's right. Did gastrin have a positive or negative effect on the stomach?
Positive?
Yeah. When is gastrin secreted by the stomach?
Gastrin. Sorry, 1 second. Gastrin is secreted by the stomach when the epithelial. When food in the stomach. When there's food in the stomach, basically.
Yeah. When there's food. Okay, that was a good answer. When there's food in the stomach, you want to stimulate the stomach to do more digestion. So, does gastrin have a positive or negative effect on the stomach?
Yeah, it's positive effect.
Right. That's why it's secreted when there's food in the stomach to increase digestion in the stomach. And do secretin and CCK have positive or negative effect on the stomach?
Negative.
Good. And why would you want them to have a negative effect on the stomach? Because when are they secreted? When are CCK and secretin secreted?
When there's food in the small intestine.
And when there's food in the small intestine, you'd want the stomach to slow down to give the food in the small intestine a chance to get digested. Okay. All right. Well, this is all stuff. Hopefully we explained this clearly, and then you can go through and work on memorizing this, again, on your own. I think these are all objectives that I think are likely to be tested. Okay. So then moving forward, that's digestion. What's the next section of the small intestine after the duodenum?
The ileum.
Right. And then what's the next section?
The jejunum.
Now, in reality, the duodenum is much, much shorter than the ileum and the jejunum. The duodenum is only, I think, like 20 centimeters long, but it looks longer on my picture because I had to have room to fit in all the words here. These are actually longer sections. So what happens in the ileum and the jejunum?
Absorption.
Yes, absorption, because digestion was basically finished where?
I'm sorry, could you repeat that?
Where was the process of digestion finished?
In the duodenum.
Yeah, the duodenum basically finishes digestion, and then absorption occurs in the ileum and the jejunum. Okay, good. So what types of things are going to be absorbed here? Well, so remember one type of molecule here was carbohydrates. So in what form will the carbohydrates be absorbed?
What form? Monosaccharides?
That's right. So here we would have absorption of monosaccharides. And the monosaccharides are absorbed into what part of the body?
Bloodstream.
That's right. Okay, then another thing we have to absorb is proteins. Well, in what form are the proteins going to be absorbed?
1 sec. Amino acids.
Right, they've been broken down to amino acids. So there'll be absorption of amino acids into what part of the body?
The bloodstream.
That's right. All right, and then the last type of biological molecule we have here is the fats. Now remember, we talked earlier, the fats have been digested into what pieces?
Into glycerol and 3 fatty acids.
Glycerol and fatty acids. And those are absorbed into what?
Those are absorbed into the lymphatic system.
Okay. All right. Now we need to talk in more detail then about this process of absorption. So absorption, so here's a picture of what's going on here. So we have, let's say, so let's say that this is the jejunum and ileum. And so this would be the lumen. This would be the epithelium. So what we want to have happen is we want all the monomers to be absorbed out of the lumen across the epithelium and into the blood or the lymphatic system. Now, there's only a limited amount of time and space for all this absorption to happen. This absorption can be a difficult and slow process. So how can we make it easier for this absorption to happen? Is the absorption going to be easier if this epithelium has a large surface area or a small surface area?
A larger surface area?
The larger the surface area, the more surface there is for these molecules to cross.
Mm-hmm.
Well, how could we increase the surface of this epithelium? Well, here's what happens to increase. That surface. What we have is we have a bunch of infoldings. So the epithelium is like it's sticking a bunch of fingers up into the lumen here. I suppose it would be doing this from both sides, but I'm only going to draw it on one side. So this is still here all the epithelial cells. The epithelial cells are tiny little cells that look like this. It would be too tedious to draw them all, but they're all lining this side here. Do you see, does this picture now have a greater or larger surface area with the lumen?
A large, I mean, there is more surface area for absorption.
Okay, good. So you didn't sound too confident about that, but yes, now there's a much larger surface area. This side over here that I've drawn the fingers on has a much larger surface bordering the lumen than before. So the purpose, these are what are called the villi. These are what are called the villi. So these are fingers that increase the surface area. And then on each of the villi, there's a bunch of little hair-like projections. Little hair-like projections pointing out, and those are called microvilli.
Okay, microvilli.
Those are little hair-like projections poking out of the surface of the epithelial cells, which give them an even larger surface area.
Mm-hmm.
So there's a huge surface area as a result of all these villi projections with their little hair-like projections on top of it. A huge surface area which makes it easier for absorption to occur because there's much more surface that the amino acids and monosaccharides, etc., can be passing over. So I think, for example, your textbook said the small intestine, if you could unfold it, would have a surface area of a tennis court. So that's kind of amazing if you stop to think about it. Obvious, so this is so extensively folded in on itself that if you actually unfolded it, it would actually spread out over a whole tennis court, even though it's so folded inside the body that it just fits inside the abdominal cavity. Okay, so the purpose of the villi and then the microvilli is to increase the surface area of the epithelium so that there's more room for the nutrients to pass across. And then inside each of these infoldings, there would be this, which is called a lacteal. The lacteal is a branch of the lymphatic system. So there would be a lacteal, which is a branch of the lymphatic system, and there would be a bunch of capillaries as well, which are branches, which are branches of the bloodstream. So the food matter could pass across the epithelial cells and then either into the capillaries or into the lacteal. If you go into the capillaries, you're entering into the bloodstream. And if you enter into the lacteal, you're entering into the lymphatic system. So the lacteal is part of what system?
The lymphatic system?
That's right. And the capillaries are part of what system?
The lymphatic system?
The capillaries are part of what system?
The circulatory system.
Okay, thanks. The blood system. That's right. And the lacteal is part of?
The lacteal is part of the lymphatic system.
Good. So what's going to be entering into the capillaries?
Blood.
Right. But what's going to be entering out of the small intestine into the capillaries? Which nutrients are absorbed into the capillaries?
Proteins and carbs.
Right. Or to be more specific, monosaccharides and amino acids. Mm-hmm. And what's going to be entering into the lacteal?
Fats.
That's right. Okay. And we still have to give a little bit more detail about what happens to those fats. So remember, these are lipids. What's the defining characteristic of a lipid? What's the definition of a lipid?
Lipids are compounds that are not soluble in water. That's
right. But the fluid inside of the capillaries and inside of the lymphatic system is water. The fluid inside of your body is water. If these are not soluble in water, how can they be carried along in the water? Well, the answer is they have to be packaged. So the glycerol and fatty acids are absorbed into the intestinal epithelial cells. Inside the intestinal epithelial cells, the glycerol and fatty acids are recombined into fats. But remember, are fats soluble in water?
No.
No. So the fat has to be packaged so that it will be soluble in water. The fat is mixed with cholesterol and coated with proteins to form what are called chylomicrons. which are soluble in water. So then the chylomicrons move out of the epithelial and into the lacteal. And remember, from the lacteal, they can move into the rest of the lymphatic system. Tell me when you have that written down.
Okay, 1 second. One second, sorry. Okay.
Okay. So remember, we'd already talked about how inside the small intestine, the lipase digests the fat globs into glycerol and fatty acids. Then the glycerol and the fatty acids are absorbed into the intestinal epithelial cells.
Mm-hmm.
These cells here that are lining the border of the lumen. And what happens to the fatty acids and the glycerol when they enter in to those epithelial cells? I got a little disorganized here. Okay, here we go. So they're recombined into fats. But what's the problem with fats? Are fats soluble in water?
No.
So the fat cannot be transported through the body unless it's packaged in a way that will make it soluble in water. So inside these epithelial cells, the fat is mixed with cholesterol, then coated with proteins to form chylomicrons, which are soluble in water. I think it's the proteins that make it soluble in water. The coating of proteins make them soluble in water. Because proteins are soluble in water. So if you coat them with proteins, then they will be soluble in water. The proteins are kind of playing a similar role that the bile salts played earlier. Just like the bile salts made it easier for the fat to move through the digestive tract. So that's what the proteins are doing here. And then the chylomicrons, then now that they're soluble in water, they can move out of the epithelial cells and into the lacteal. So we have the glycerol and fatty acids that move out of the lumen into these little epithelial cells. And the epithelial cells, they're repackaged as chylomicrons, and then the chylomicrons move into the lacteal. Okay. So remember that lipase digests fat droplets into glycerol and fatty acids. Then what happens to the glycerol and fatty acids?
The glycerol and fatty acids move into the into the villi, into the lacteal.
Good, but that's skipping over a bunch of steps that I think you might be tested on. So we need to talk about those steps. So the glycerol and fatty acids were formed inside the lumen of the small intestine, and then where do they move?
They move through the
let's take a while to actually just memorize this. So here's the key steps. This is a little complicated, so take your time and try to memorize these steps and tell me when you're ready to keep going.
Okay. Okay, so absorbed into the intestinal epithelial cells inside the intestinal epithelial cells, the glycerol and fatty acids are recombined into fats. The fat is mixed with the cholesterol and coated with proteins to form chylomicrons.
Good.
Chylomicrons, which are soluble in water. The chylomicrons move out of the epithelial cell into the lacteal.
And you should be trying to visualize what's happening there. It's actually not that you should be visualizing what's actually happening. The glycerol and fatty acids were formed in the lumen. They move into the epithelial cells here. And then before they can move out of the epithelial cells, they're repackaged in fat, which is repackaged in chylomicrons. And then the chylomicrons move to the lacteal. Okay, well, if you think you have that down, what happens to the glycerol and fatty acids after they've been formed inside the lumen?
I'm sorry, your mic is breaking up again.
What happens to the glycerol and the fatty acids after they've been formed inside the lumen?
After they're formed inside the lumen, the glycerol and fatty acids are recombined into fats. The fat...
Where does that happen?
That happens... Wait, the epithelial cells... that happens when the fat is mixed with the cholesterol and it's coated with the proteins to form chylomicrons.
Great. Where is that happening? Where is that process happening?
In the epithelial cells.
Of the small intestine. Good.
Yeah. So the fats, this is all happening in the epithelial cell inside the small intestine. The glycerol fatty acids are recombined into fats. The fat is mixed with cholesterol and coated with proteins to form chylomicrons, which are soluble in water.
Good.
The chylomicrons, they move out of the epithelial cell into the lacteal.
Good. Okay, good. So let's go through that one more time. So what happens to the glycerol and fatty acids after they're formed in the lumen?
After they're formed in the lumen, they're absorbed into the intestinal epithelial cell, and the glycerol and fatty acids are recombined into fats.
Good.
The fat is mixed with cholesterol and coated with proteins to form chylomicrons, which are soluble in water. The chylomicrons move out of the epithelial cell into the lacteal.
Okay, good. And the lacteal is part of what body system?
The lymphatic system.
Okay, that's right. Okay, very good. So, good. So, how would you describe how is the the surface of the small intestine has extended foldings and finger-like projections. What's the function of all that folding and those projections?
To increase the surface area space.
Excellent. Right. To increase the surface area that borders the lumen. To increase the surface area that borders the lumen. How does it help to have how does it help to have a greater surface area?
I'm sorry?
Why is it helpful to have a greater surface area?
The more surface area, the more the glycerol and fatty acids have more room to absorb. Like more of these molecules can be absorbed.
That's right. Remember, anything that doesn't get absorbed in the small intestine is just going to be eliminated in the feces, and it's not going to do the organism any good. So we want to give maximum chance for as many nutrients as possible to be absorbed. So we need a high surface area to increase the area that's available for absorption, not only of glycerol and fatty acids, but also of amino acids and monosaccharides. Good. Do you remember what do we call these projections in the wall of the small intestine?
Microvilli?
These are the villi, and then the hair-like projections are the microvilli.
Okay.
Okay, good. All right, so, and where is this absorption happening?
In the epithelial of the small intestine.
Yeah, which part of the small intestine?
The lumen?
Well, which part of the small intestine? The ileum and the jejunum is what I was going for. Okay. All right. So then what's going to happen? Where does the food that didn't get absorbed pass next?
Passes through the large intestine.
Okay, good. What's the function of the large intestine?
Mainly absorption of water.
Yeah.
And whatever molecules haven't been digested.
Now, that last thing you said didn't quite make sense. If things haven't been digested, they can't be absorbed. You said there would be absorption of water and things that haven't been digested. But do you see how that doesn't make sense? If something hasn't been digested, it can't be absorbed. So what's the other function here besides absorption of water? The other absorption is what happens to the stuff that hasn't been absorbed? It hasn't been digested. That just gets formed into the feces. So absorption of water and formation of feces from undigested food matter. For example, do you remember cellulose can't be digested from plant cell walls? So that is going to enter into the feces. So formation of feces from undigested food matter is something else that's happening here in the large intestine. Okay. By the way, something else that is worth mentioning, that lots and lots of bacteria live in the large intestine. You might have heard of E. coli. E. coli is the type of one of the types of bacteria that live in the large intestine. And we usually think of bacteria as causing disease. But these bacteria are usually not harmful. In fact, oftentimes these bacteria can be helpful to the organism because the bacteria could digest some of the food matter that didn't get digested by the organism itself. And sometimes the bacteria are going to digest some of this food matter and they're going to form vitamins that can still be absorbed by the large intestine. So we might make a note. The large intestine has the function of absorption of water, and also it absorbs some vitamins that are produced by bacteria in the large intestine, digesting the food that didn't get digested before. Okay, so the functions of the large intestine are absorption of water, formation of feces. Lots of bacteria live in the large intestine, and they can further digest some of the food and create vitamins, which could also be absorbed out of the large intestine. Also, sometimes when those bacteria are digesting this food matter, sometimes as part of their digestion, the bacteria form gases. And those are the gases that can cause flatulence. So that's another effect of the bacteria in the large intestine. And then at the end of the large intestine, we have the rectum, where the feces are stored, and then the anus, where the feces are eliminated. So now we've gone through the complete sequence of the digestive tract here. So what would be the hormone that, what's the hormone, describe the hormonal control of the stomach.
The hormonal control of the stomach, I guess it would be, there would be a negative effect. The CCK and the secretin would cause the stomach to slow down digestion.
When would that occur?
That would be when the food passes through the stomach and into the small intestine.
Good. Good.
So
now there's also another hormone that that the stomach releases. What's the hormone that's released by the stomach? Gastrin. When is that released?
When there is food matter in the stomach.
That's right. And does that have a positive or negative effect on the stomach?
Positive.
Good. And then what would be another target of CCK besides the stomach?
The gallbladder.
Good. And what does it cause the gallbladder to do?
To release the bile.
Good. And what's another target of CCK besides the gallbladder and the stomach?
Pancreas.
And it causes the pancreas to?
Release the enzymes.
Good. And then another enzyme besides CCK is, and gastrin is?
Secretin.
Good. And what's its target?
It targets the pancreas to release the bicarbonate.
Right. And the stomach as well, as you already mentioned. Good. What's the function of the bicarbonate?
One second. Sorry. The bicarbonate, it's released into the duodenum to neutralize the HCl from the stomach.
Excellent. Good. So what does it do to the pH?
It increases the pH.
Okay. Very good. Excellent. That's right. Okay. Good. All right. I think there's one more key topic we need to go over in this chapter, which is glucose regulation. This is usually tested. So this is a little bit of a separate topic. Do you know what type of molecule is glucose?
Glucagon?
The question is, what type of molecule is glucose?
Oh, sugar.
Yeah, it's a type of sugar. That's right.
Yeah.
So it's a type of carbohydrate.
Yeah, carbohydrate.
To be more specific, it's a monosaccharide.
Okay.
Glucose is a monosaccharide, maybe the most important monosaccharide. It's a monosaccharide that you probably focused on in the first part of the course when you were going over cellular respiration. So glucose plays a big role in the Krebs cycle, for example, and in glycolysis, which you studied in the first semester. Okay,
Yeah.
so remember that in chapter 40, which we haven't talked about very much, there's this concept of homeostasis, which is that the body tries to keep all of its various levels and concentrations and conditions at steady state levels. You need to keep the amount of every solute in the blood at a pretty constant level. If there's too much of any substance, that will cause a malfunction in the body, and if there's too little of any substance, that will cause a malfunction in the body as well. So one thing that the body has to do is maintain the right levels of blood glucose, the right amount of glucose in the blood. So we need to go over the control of the amount of blood glucose in the blood. So suppose that blood glucose levels rise. Remember what type of molecule is glucose?
Take monosaccharide.
That's right. So, what should the body want to do if blood glucose levels are too high? This is a kind of simple question. What does it want to do?
Yeah.
Does it want to now increase or decrease glucose levels?
Decrease.
Decrease them. Yes. So let's see how the body is going to do that. So when blood glucose levels rise, that causes the pancreas to secrete insulin. We already talked about the pancreas many times in this chapter, so here's a whole completely different role of the pancreas. This is just a completely different role of the pancreas than we talked about before. The pancreas is going to secrete insulin. Insulin is another hormone. So we've seen the pancreas secretes a lot of hormones. Remember that hormones carry messages to other parts of the body. So now we have to see what is the insulin going to cause the other parts of the body to do. So then the insulin causes body cells to take in glucose. Do you see how this is helpful? If the body cells are taking in more glucose, what will happen to the amount of glucose in the blood?
If the body cells are taking in more, I'm sorry, what?
If the body cells are taking in more glucose, what will happen to the amount of glucose in the blood?
It'll increase.
Did you say increase or decrease?
Increase.
I think we're not thinking about that correctly. If you're taking the glucose, so if you're taking the glucose into the cells, you're taking it out of the blood. Is the point. The body cells are taking in glucose, so that leaves less glucose left over to be in the blood. So the idea here is that the glucose is being taken out of the blood and into the body cells. So what's this? So when the body cells take in more glucose, what's going to happen to the amount of glucose in the blood?
It's going to decrease.
Which is what we wanted, right, to get back to homeostasis. Yeah. So the insulin will cause body cells to take in glucose. Also,
okay.
The insulin also causes glucose to be stored as glycogen in the liver and muscles. So here's a glucose. Here's a glucose. Here's a glucose. Here's a glucose. Glycogen is a bunch of glucoses connected to each other. So if you take a bunch of glucoses and connect them to each other, you would get glycogen. You can tell that the name glycogen sounds like glucose to remind us that it's made out of a bunch of glucoses. So what type of molecule is glucose?
And glucose is a monosaccharide?
That's right. So what type of molecule is glycogen?
Polysaccharide?
That's right. You can see from the picture here, glycogen is a polysaccharide. It's a way of storing the glucose. I don't know if you remember, but in the previous sessions we mentioned glycogen is a way that animals store energy. Glycogen is a polysaccharide for animals storing energy. Well, that's what we're doing here. There's more glucose than we need in the blood. So we need to store that energy for later in the liver and the muscles. So that's going to be stored as glycogen in the liver and in the muscles. Now, if the insulin is causing glucose to be stored as glycogen in the liver and the muscles, what's going to happen to the amount of glucose in the blood?
The glucose in the blood is going to decrease.
That's right. Well, remember that was what we wanted to accomplish all along. So would this be called positive or negative feedback?
Positive.
This is called negative feedback because the increase in blood glucose was canceled out.
Okay.
Blood glucose went up, and then that caused the body to respond in ways that would decrease blood glucose. So this is a negative feedback loop because the body is trying to cancel out the increase in the blood glucose levels.
Okay.
Okay.
But I feel like, isn't it doing what we needed to do?
It is. So when we say negative feedback, we don't mean negative like bad. We don't mean that it's bad. We just mean that just like, you know, when you do math, negative numbers are not bad, evil numbers. They're just different from positive numbers. So negative feedback is not a bad, evil type of feedback. It's actually usually the right type of feedback because that's the way to have homeostasis, right? Homeostasis means everything has to be kept at a fairly steady level. So that means whenever anything increases, the body needs to decrease it to get it back to the previous level. That's negative feedback. So negative feedback is the normal way that the body maintains homeostasis. Negative feedback is the normal way the body maintains homeostasis. When something increases, the body responds to that in it. So maybe instead of calling this negative feedback, we might call it opposing feedback or canceling feedback. We're trying to cancel the previous effect. That make any sense?
Yeah, that makes more sense.
Okay. So let's study this sequence here. I think this is pretty popular on exams. So remember that what started this is when blood glucose levels increased. What does the body want to do when blood glucose levels increase?
The body wants to release insulin to cause the body cells to take in the glucose and to lower the blood glucose levels.
Yeah. What I was going for is the body wants to get back to homeostasis by decreasing blood glucose levels, and then we can go through how does it do that. So how does it respond? So when blood glucose levels rise, what's the first response in the body?
To release insulin?
From
The pancreas.
Good. What type of molecule is insulin?
It's a monosaccharide.
Actually, I think you're getting a little confused there. Try again. That's glucose. Glucose is a monosaccharide. What type of molecule is insulin?
It's an enzyme.
I'm not sure if it's an enzyme or not. It's a hormone.
Hormone.
Because it's sending a message, right? It's transporting the message that's going to cause other things to do other things. So I don't think it's an enzyme, but it is a hormone. All right. And then what message does the insulin carry? What does the insulin do?
The insulin causes the body cells to take in glucose.
Good. What does that do to the blood glucose levels?
Which will decrease blood glucose levels.
Because if there's more glucose in the cells, there's less glucose left over for the blood. Good. And what else does the insulin cause?
The insulin also causes glucose to be stored as glycogen in the liver.
And what would that do to blood glucose?
It would also lower it.
Right. Because I think that's pretty obvious. Okay, so stare at this for a few seconds. Tell me when you feel you have that memorized.
Okay.
Okay, so let's say that blood glucose levels increase. Describe the series of steps that would happen next.
As blood glucose level increases, it needs to maintain homeostasis. So the pancreas will secrete insulin, and that insulin will cause the body cells to take in the glucose, which lowers the blood glucose levels. But also, the insulin also causes the glucose to be stored as glycogen. So the glucose stores as glycogen in the liver, and the liver and the muscles.
Right.
That all.
Good.
I believe that's all.
And then what does that do to blood glucose levels?
Oh, that also lowers blood glucose levels?
So would this be an example of positive or negative feedback?
Positive.
Did you say positive?
Wait, do we say... negative feedback?
Yeah, maybe we should review those terms. So let's say that an increase in A causes more increase in A. That would be positive feedback. Let's say that an increase in A causes a decrease in A. That would be negative feedback. Which of these types of feedback is more useful for maintaining homeostasis?
Positive.
Actually, that's a misunderstanding. Remember that homeostasis means that you want the level of A to be constant. So positive feedback would be the exact opposite of what you wanted, right? So for example, suppose that A started at 10 and increased to 11. If there was positive feedback, it would increase even more to 12. And then the positive feedback would increase it to 13. And then the positive feedback would increase it to 14. And then the positive feedback would increase it to 15. Notice we're getting away from the original level. We're not getting back to the original level. Positive feedback destroys homeostasis. Instead, let's say that you started at 10, and then it increases to 11, and then there's that decreases back to 10. That would get you back to, that would be negative feedback, right? When an increase in A causes a decrease in A. So almost all of the feedback loops in the body are negative feedback. There are some positive feedback loops, but they're very rare. There are some positive feedback loops in the body, but they're very rare because they tend to destroy homeostasis. The large majority of feedback loops are negative feedback because that's the way to get back to homeostasis. Do you see the difference between these?
Mm-hmm.
Okay, so this system of insulin, is insulin part of a positive or a negative feedback loop?
So it'll be a negative feedback loop?
Because the original increase in blood glucose causes a series of steps that cause a decrease in blood glucose. Okay. Mm-hmm. All right, that's right. What type of molecule is insulin?
Insulin is a hormone.
Good. What type of molecule is glucose?
Glucose is a carbohydrate.
Good. What type of carbohydrate?
Monosaccharide.
That's right. And what type of molecule is glycogen?
Polysaccharide.
That's right. It's the way that the glucose is stored. When would blood glucose levels rise? Well, blood glucose levels tend to rise, say, after you've eaten, right? If you just ate, you would eat a lot of carbohydrates. The carbohydrates would get digested, and then they would get absorbed. And so that would add a bunch more glucose to the blood, right? Mm-hmm. So when does insulin tend to be released? It tends to be released after meals.
Yeah.
Insulin tends to be released after meals. The insulin, the pancreas says, "Oh, it looks like the person just ate another meal. We need to keep all that extra. We have to do something with all this extra glucose. We need to store the glucose in cells or in the liver or in the muscles. Remember, glycogen is an energy storage molecule. So, for example, when you're exercising, you might need extra energy, and your muscles might start breaking down glycogen back into glucose to give the muscles extra energy. So it makes sense that glycogen would be partially stored in the muscles, because that's a source that oftentimes uses up its energy sources. Okay, well, there's a whole other side to this, which is what happens when blood glucose levels decrease. Now, when blood glucose levels decrease, first of all, what would the body want to accomplish then?
So blood glucose, the body would want to increase glucose levels?
To get back to homeostasis. That's right.
Yep.
So let's see how the body accomplishes that. Well, once again, the organ that responds is the pancreas. But the pancreas can't respond to this with insulin, because insulin would have the exact wrong effect right now. So the pancreas now has to secrete a different hormone. Now the pancreas secretes glucagon.
Okay, let me write that down.
Glucagon. It's called glucagon to remind us that it's related to the regulation of glucose. So glucagon is a hormone.
Yep.
Okay. All right, are you still writing? Are you ready?
Still ready.
Okay, yeah, take your time.
Okay.
What type of molecule is glucagon?
Glucagon is a hormone.
That's right. You don't want to confuse it with glucose or glycogen.
Yeah.
So all these names sound similar, but they're different things. All right, so what is the glucagon going to do? So the glucagon is going to go to the liver. Now, what would the body want to do now? Would it want to turn glucose into glycogen or turn glycogen into glucose?
Could you repeat that question? I'm sorry.
What would the body want to accomplish now? Would it be helpful now to turn glycogen into glucose or to turn glucose into glycogen?
It would be better to turn glycogen into glucose, since we're trying to increase glucose levels.
Excellent. That's the exact right logic. That's the exact right logic. So glycogen, so the pancreas causes the liver to break down glycogen. When glycogen is broken down into pieces, what are the pieces called?
Glucose molecules?
That's right. It's made up of glucose. So the glycogen is broken down into glucose. And the glucose is released into the blood. So when you're ready, what would that do to the blood glucose levels? Take your time.
Increase.
Good. Is that what we wanted?
Yes.
Yeah, so would this be positive or negative feedback?
Positive feedback? No, wait. No, negative. No, wait, hold on. Okay, so positive is when A goes up, and then that causes A to go up. Negative feedback is when when something goes up and another thing goes up. So
I think we're still getting confused about that, so let's go back and review that. Positive feedback is when A goes up, and that causes A to go up even more. Negative feedback is when A goes up, and that causes A to go down. So it's the same thing in both cases. When something increases, and that causes that same thing to increase even more, that's positive feedback. When something increases, and then that causes that same thing to decrease, that's negative feedback. Do you see why this would be called negative feedback, and this would be called positive feedback?
Mm-hmm.
Okay. So what did we have here? At the start, blood glucose levels decreased. And then what happened to the blood glucose levels at the end of the sequence?
The blood glucose levels increase.
So would that be positive or negative feedback?
Negative.
Right. Maybe you said positive because you thought the word increase was a positive sounding word. But the point here is that the glucose was moving in two different directions. The glucose first decreased, then increased. So whenever the first change is being canceled out, that's negative feedback. Maybe we should specify that a little bit more. So, is this positive or negative feedback?
Positive. I mean, yes, it's positive.
Is this positive or negative feedback?
Negative.
Now, we haven't quite talked about this before, but we should be able to figure this out from the earlier examples. Is this positive or negative feedback?
Positive?
Yeah, it's positive feedback. You might think it was negative because things were decreasing, but the point is, if the change in A causes another change of the same type, that's called a positive feedback loop. And what type of feedback is this?
Negative.
Because here, the original change in A caused an opposite change in A. So when a change causes another change of the same type, that's positive feedback. But when a change causes a change of the opposite type, that's negative feedback. So what type of feedback did we have in this case? First, blood glucose levels decreased, and then what happened to the blood glucose levels at the end?
Increased.
So what type of feedback was it?
Negative.
Remember that almost all feedback loops in the body are negative. Almost all feedback loops are negative because that's the only type of loop that can maintain homeostasis. Positive feedback does occur, but it's actually quite unusual. Okay, so this is the whole sequence here for glucagon. It's a little bit simpler than for insulin. So again, it would be good to study this and try to memorize this. So blood glucose levels decrease. What happens next?
Once the blood glucose levels decrease, the pancreas will secrete glucagon.
Good, and then what?
And then the liver, the glucagon, wait, will the glucagon travel into the liver?
That's right. So what type of molecule is glucagon?
It's a hormone.
A hormone. So the pancreas will secrete glucagon into the bloodstream. And then the bloodstream will carry glucagon to the liver. And then the glucagon will...
The glucagon is broken down into glucose. And
Say
then
that one more time, please.
The glucagon will cause the liver to create glycogen.
That still wasn't quite right. The glycogen's already there. It doesn't have to be created.
So the glucagon will cause the liver to break down glycogen, and then the glycogen will break down to glucose.
Right.
Then the glucose is released into the blood.
Okay, that's right.
And then the blood glucose levels increase.
That's right. Those are two parts. That's right. Okay, so what type of molecule is glucagon?
Hormone?
And what type of molecule is glycogen?
Glycogen is oh, hormone.
We should review that. Remember this table here? Glycogen is a polysaccharide that's made of glucose monosaccharides. Remember that glycogen is an energy storage molecule. When the body wants to store energy, it stores it as glycogen.
All right, so I'll write that. Okay.
So what type of molecule is glycogen?
Polysaccharide.
So in reality, glycogen would be made of, well, what type of molecule? Glycogen is made of what?
Multiple glucose.
Yeah.
A chain of glucose.
A chain of glucose. In reality, it would be made of hundreds or thousands of glucoses. What type of molecule is glucose?
Glucose is a monosaccharide.
Right. And what type of molecule is glucagon?
Hormone.
That's right. And what does the glucagon do?
The glucagon causes the liver to break down glycogen. And then the glycogen will break down into glucose. Then the glucose is released into the blood, and that causes increased blood glucose levels.
Okay, that's right. So take a few seconds and try to memorize this sequence.
Okay.
Okay, good. So, how does the body respond when blood glucose levels decrease?
1 second.
Yeah, take your time.
So once blood glucose levels decrease, the pancreas will secrete glucagon. That glucagon goes into the liver, and then it allows glycogen to be broken down into glucose and then released into the blood. Yeah, and then once it's released into the blood, it causes the blood glucose levels to increase.
Okay, excellent. Very good. How would the body respond if blood glucose levels increased?
Oh wow. 1 second.
So these are popular types of test questions.
Yeah, if blood glucose levels increased, we would need to maintain homeostasis and decrease blood glucose levels. So insulin would be released into the body, into the body cells, I believe, and the body cells would take in the glucose, removing it from the blood.
Excellent.
In order to decrease the blood glucose levels.
Excellent.
Okay. I'm forgetting the last part if there is one.
So what would happen with glycogen and glucose? would you want the glycogen to form into glucose or glucose to form into glycogen in this situation?
The glycogen to form into glucose.
Like this?
I guess the insulin will cause, no, because that's what happens when there's low. So would you want the glucose to become the glycogen?
So this is something we should be able to figure out. This is something we shouldn't have to memorize. We should be able to figure it out. Which sequence would give us the desired outcome?
Glucose into glycogen.
That's right, because what will that do to blood glucose levels?
Decrease it. And
that's what we wanted, right? So a lot of this is much easier if we understand the logic behind it. That way we don't have to rely only on our memory. We shouldn't have to memorize whether we're going from glucose to glycogen or glycogen to glucose. It should make sense based on what the body is trying to accomplish to get back to homeostasis.
Yeah.
Okay, so I think we worked this out correctly. So you have to memorize that insulin is released when glucose goes up and glucagon is released when blood glucose goes down. But we shouldn't have to memorize whether the glucose is changing into glycogen or the glycogen is changing into glucose. That's something that we can figure out. Okay, so suppose that blood glucose levels increase. How would the body respond?
It would release insulin. And the insulin would cause the glucose to become glucagon. Or no, I mean glycogen.
Right. It's important to try not to confuse the names of these molecules. Good. Good. And that would lower blood glucose levels. Excellent. Let's say that blood glucose levels decreased. What would that cause to happen?
1 second.
Yeah, take your time.
Once blood glucose levels decrease, the pancreas will secrete the glucagon. The glucagon will cause the liver. The glucagon will go into the liver, and the glycogen will be broken down into glucose, and then it will be released into the blood. Once the glucose is released into the blood, it causes the blood sugar levels to rise.
So would that be positive or negative feedback?
Negative.
Which is what we want for homeostasis, right?
Yeah.
Remember that you need negative feedback loops to maintain homeostasis. Okay, good. So when blood glucose levels decrease, what hormone is released?
When blood glucose levels decrease, glucagon is released.
Good. And when blood glucose levels increase, what hormone is released?
Insulin.
All right, so those are ideas that have to be memorized for the test. I think those are pretty likely to be tested. Okay, so let's see where we're at here. So it takes a long time to go over all of these objectives. This first objective here we haven't really talked about, but I think this is a pretty simple one for you to learn on your own. We haven't really talked about the second objective, but I think it's a pretty simple one to learn on your own. Here is what we've been working on. So how would you describe the homeostatic regulation of glucose?
I guess the homeostatic regulation of glucose. All right. Could I say in order to sustain homeostasis of glucose, the pancreatic hormones, insulin and glucagon.
Good. So the best way to answer that type of question would be to give specific cases. So for example, what would the body do if blood glucose levels increased?
So, okay, if the blood glucose level increased, insulin would be released. If blood glucose level
now
decreased,
let's follow up that sequence. You'd have to give more details. So what would be the result of the insulin being released?
When the insulin is released, the glucose will turn into the glucagon and lower the blood glucose.
Take your time. The glucose will turn into what?
Glycogen.
Right, not glucagon. Okay, good. Where will that occur?
In the in the bloodstream?
So actually, the those are
in the
formed in
liver?
the liver and the muscle cells.
Okay, yeah. In the liver.
The glucose is stored as glycogen in the liver and the muscle cells.
All right.
So take your time. I think there's a good chance this will be on the test. So when blood glucose, so let's take our time and describe it one more time. When blood glucose levels increase, how does the body respond? When blood glucose levels increase, how does the body respond?
When blood glucose levels increase, the body responds by releasing insulin, and then that insulin causes the glucose to become glycogen, and that causes.
And where does that occur?
In the liver and the muscle.
Good. Remember, it makes sense that the muscles would be storing energy because they need energy later on. Okay, good.
And then that causes a decrease in blood glucose levels.
Good. And remember, the other thing the insulin does is it causes the body cells to take in glucose. That would also decrease blood glucose levels. Tell me when you're ready.
Blood cells take in glucose.
Body cells.
Yeah.
So this we're just reviewing what we already talked about, right? So here's what we talked about. The pancreas secretes insulin. That causes the body cells to take in glucose and also for glucose to be stored as glycogen in the liver and the muscles.
Okay.
So then what happens when blood glucose levels decrease? How does the body respond to that?
Once blood glucose levels decrease, the pancreas secretes glucagon. The glucagon goes into the liver, and then the glycogen is broken down into glucose and released into the blood. This causes the blood glucose levels to increase.
Good. So would that be positive or negative feedback?
Negative.
So that gets us back to homeostasis. Okay. Well, that's something you can review on your own. I think there's a good chance that would be tested. That's a popular topic. Here's this objective. Define and compare the four main stages of food processing. So what are the four main stages of food processing?
Ingestion, digestion, absorption, and elimination.
That's right. What does digestion mean?
The turning monomers into breaking down of monomers into polymers.
Say that one more time, please.
The breakdown of polymers into monomers.
Very good. Excellent. Good. So that we want to be careful about that on a test answer. Good. And then define absorption.
Absorption is basically the digested material leaving the small intestine and going to either the bloodstream or the lymphatic system.
That's right. Now, to be more specific, it would be the monomers here that would be absorbed. Also, you might not want to use the term small intestine here because part of this chapter is about the human and mammalian digestive system, but part of the chapter is about animals in general. Not all animals have a small intestine, but they all have to do absorption. So this question here is not necessarily about mammals in particular. So what does absorption mean in general? Absorption means digestion is when the polymers are broken down into monomers, and then absorption is when the monomers are brought into the body cells. Okay, this next objective we haven't really talked about too much. This objective we talked about, describe the common processes and structural components of the human digestive system. Well, the structural components are things like the mouth, esophagus, stomach, small intestine, large intestine, the pancreas, the salivary glands. So this would be too much for us to review right now, but this is kind of one of the main things we've been spending our time on, all these different processes that we talked about. We haven't really talked much about this. We spent a lot of time on this objective, where and how the major types of macromolecules are digested and absorbed. So where are carbohydrates digested?
In the mouth and in the small intestine?
That's right, particularly in the duodenum. And where are they absorbed?
They're absorbed into the ileum and then the jejunum.
They're absorbed out of the ileum and the jejunum. Good.
Yeah.
And where are proteins digested?
In the stomach and the small intestine.
That's right. And where are fats digested?
The small intestine.
That's right. All right. Explain the absorption of nutrients in the small intestine and the absorption of fats. That's something we spent a bunch of time on today. So which sections of the small intestine are used for absorption?
The ileum and the jejunum.
That's right. Okay, good. And the amino acids and monosaccharides are absorbed into what?
The amino acids and monosaccharides are absorbed into the, did you say the bloodstream?
Into the bloodstream, yes.
Yeah, I wasn't sure if you wanted to.
And where are the fats absorbed into?
The lymphatic system.
Right. Let's be a little more specific about that. So can you give me more details about the absorption of fats? So remember, the fats are broken down into glycerol and fatty acids inside the lumen of the small intestine. And then what happens?
So basically, you want me to describe the digestion of fats?
Well, we've already talked about their digestion. So they've been digested now into glycerol and fatty acids. After they've been digested, what happens after they've been digested? How are they absorbed?
They're absorbed out of the small intestine into the lymphatic system.
Remember, we went into a lot of details about that today. So what were the details of that process?
Okay. So the glycerol and fatty acids are absorbed into the small intestine and into the intestinal epithelial cells inside the. Sorry.
That's good.
The glycerol and fatty acids recombine into fats.
Good.
The fats are mixed with the cholesterol and coated with protein to form chylomicrons.
Good.
Those are soluble in water.
Good.
So the chylomicrons, they're able to move out of the epithelial cell and into the lacteal.
Excellent. And the lacteal is part of what?
The ileum and the jejunum.
Let's see. I think that would be a mistake. No.
Well, this is happening in the ileum jejunum.
Well, they've exited the ileum and the jejunum, and the lacteal is part of what? The lacteal delivers them into what? What I was going for is it's part of the lymphatic system.
Okay.
So the lacteal is a vessel of the lymphatic system. Capillaries are vessels of the bloodstream, the blood system, circulatory system, and lacteals are vessels in the lymphatic system.
Okay.
So what are lacteals part of?
Vessels in the lymphatic system.
Right. And what are capillaries part of?
The the circulatory system.
Good. Okay. Well, that's something you can review more on your own. The next objective is, that's pretty good recollection for now, though. Explain how the small intestine is specialized for digestion and absorption. Well, we especially focused on absorption. What do you think they're going for here? How is the small intestine specialized for absorption? How is the small intestine specialized for absorption? What are they talking about?
They're talking about how they have, since the small intestine has two other parts just for absorption, the ileum and the jejunum.
Good. They're going for something else though here. How is the small intestine shaped to help absorption?
I'm not sure what you mean by that.
So what they're going for here is, remember we talked about the villi and the microvilli.
Yeah.
How the wall of the small intestine is folded in
Oh, the
That's what they're
surface
talking about here. That's
Okay.
what they mean when they want you to explain how the small intestine is specialized for absorption. They want to talk
Okay.
about how it's shaped for absorption. So how do those
Yeah.
villi and microvilli help the process of absorption?
It helps the process for absorption because it allows a larger surface area.
Excellent. That's the key. So they're
Okay.
specialized for absorption by having a very large surface area. Remember, the textbook said that if you actually unfolded them, they could cover an entire tennis court. So
Mm-hmm.
they have a very high surface area. Okay, well, that's something else, again, that you can review on your own. The next objective, describe the major functions of the large intestine. The next objective is describe the major functions of the large intestine. How would you answer that if that was a test question?
The major functions of the large intestine would be the absorption of water and the formation of feces from the undigested food matter.
Good. You might also mention, remember that there's many bacteria that live in the large intestine. They can digest some of the undigested food and produce vitamins, and the large intestine also absorbs some of those vitamins as well. So the large intestine absorbs water and vitamins. Let's see. Number 12, we really haven't talked about very much, but it's in the notes if you want to review that. Number 11, we spent some time on today. Now, we only did part of this. So describe the role of hormones in regulating digestion. So we spent a lot of time on that. So when food enters the stomach, what's the hormonal response?
When food enters the stomach, the hormonal response will be to break down the food, so
Well,
HCl
what hormone is released?
and pepsinogen.
Now, careful, neither of those are hormones.
Oh,
What hormone is released when food enters the
Gastric.
stomach?
Gastric. I'm
Gastrin.
sorry.
That's right. That's okay.
Yeah.
So
I'm
gastrin
thinking.
is released by the stomach, and then the gastrin causes the release of the pepsinogen and the hydrochloric acid. Okay?
Yes.
That's good. Then what happens when the food enters into the small intestine? What's the
When the
hormonal
food,
response to that?
the hormonal response will be CCK and secretin to be released.
Mm-hmm. And what do they do?
The targets of CCK would be the gallbladder and the pancreas, but also the stomach.
Mm-hmm.
But the gallbladder and the pancreas have a positive feedback. And for the stomach, it's a negative feedback because the CCK needs to slow down the CCK needs to slow down digestion in the stomach. So CCK and secretin will cause a negative feedback in the stomach to allow the small intestine to catch up. And then
Good.
the CCK will target the gallbladder to release the bile.
Good.
And then it'll also target the pancreas to release the enzymes. The secretin will target the pancreas in order to release the bicarbonate.
Good. Good. Okay. Good. All right. Now, what do they mean here? The role of hormones in energy storage. I think here they're talking about the blood glucose levels again. So if blood glucose levels were high, would that be an indication that the body has lots of energy, has plenty of energy stores, or is running out of energy? If blood glucose levels are high, does that mean that the body has lots of surplus energy or a deficit of energy?
A surplus of energy?
Yeah, because there's all this glucose floating around that the body doesn't need. So when there's blood glucose levels are high, there's kind of surplus energy. So how would the body respond to having all that surplus glucose floating around? What hormone is released?
When blood glucose levels are up, insulin will be released.
That's right. And that causes the glucose to be stored as what?
Glycogen.
So notice that is a way of storing energy, is the point. When blood glucose levels are high, that causes the body to store energy as glycogen. Now, what would it mean if blood glucose levels were very low? If blood glucose levels were very low, would that be an indication that the body has a surplus or a shortage of energy?
Shortage.
Yeah, because it doesn't have enough glucose to run glycolysis in the Krebs cycle. So what hormone would be released then?
The pancreas will secrete glucagon.
That's right. And then we need to get some more energy. So the glucagon, where's the glucagon going to go to get the extra energy we need?
It's going to go into the liver.
Yeah, and where will the liver get the energy?
If from the okay, 1 second. The glycogen is broken down into glucose and released into the blood.
And then that glucose can be taken into cells and used in cellular respiration to be made into ATP for energy, as you learned in the first semester. Okay, so I think this energy storage, role of hormones in energy storage is pretty much the same objective as this one up here, number 3, homeostatic regulation of glucose. I think there's a good chance that would be tested. That's a popular topic. They also mention here the role of hormones in regulating the appetite. We didn't talk about that together, but you can memorize those hormones if you think those are going to be tested. So what does... what does this show happening?
This basically shows pepsin, HCl being released, pepsinogen activating, becoming pepsin. And then it also shows the pepsin activating more pepsinogen and turning that into pepsin. So it's basically a cycle.
Good. Also, notice one more thing. Notice how the pepsinogen came from these red cells, but the HCl came from these yellow cells. They came from 2 different cells. The pepsinogen came from these chief cells. The HCl came from these parietal cells. Remember that they have to be produced in separate places so that they don't react with, so that the pepsinogen doesn't get activated. They don't meet each other until they reach the lumen of the stomach, and that's where this process occurs. I'm not sure whether your instructor is going to test you on these names here, chief cell and parietal cell. You should know they're 2 different cells. Okay, good. So we talked, this is some of the key elements here. The acid chyme exits from the stomach into the first section of the small intestine, which is called the duodenum. And notice that the liver feeds bile into the gallbladder, which stores the bile and feeds it into the duodenum, and the pancreas also delivers into the duodenum. We've also learned that the pancreas does other things that are not involved with the duodenum, though, right? Remember, the pancreas also delivers insulin and glucagon into the bloodstream. That's a completely different function. Here again is a table that summarizes the enzymes that are used at every step. The only ones we went over were salivary amylase, pancreatic amylase, lipase. I don't know how many of these other ones are going to be tested or not, but they're all here in the table if you wanted to try memorizing them. Pepsin we talked about. Something we haven't talked about is, so we talked about how pepsin is secreted in an inactive form. Well, that's actually true of all the digestive enzymes. All the digestive enzymes, or almost all of them, or many of them anyway, are secreted in inactive forms. So that also happens in the small intestine. So here's some enzymes that are secreted by the pancreas into the small intestine. Notice that we haven't talked about this, but trypsin is secreted as trypsinogen. And then when it reaches the small intestine, it's converted into trypsin. And then the trypsin helps to convert this procarboxypeptidase into active carboxypeptidase. And it also activates the chymotrypsinogen into chymotrypsin. So hopefully you don't need to know all these details, but you should know that it's normal for the digestive enzymes to be created in inactive forms, and they're not activated until they get into the lumen. Why would we want the digestive enzymes to be created in inactive forms?
So they won't. So their active forms doesn't interfere with, like if pepsinogen. I don't really know how to explain it. Oh, it won't interfere like where it's located before it's released.
Except what would be a better word than interfere? To be more specific, what do these things do? They digest things, right? Well, also, you don't want them to digest you, basically.
Yeah, basically.
You don't want to digest yourself. So you want them to be created in inactive forms so that they don't digest the cells that they're created in. And then they don't become activated until they're secreted into the lumen of the digestive tract. Remember we talked about last time how a biologist thinks of the digestive tract as being actually outside of your body, like the whole of a donut is outside of the donut. So as long as these enzymes are inside of the body cells, they have to be in inactive forms, because otherwise they'll digest and destroy your own body. It's only when they're secreted into the donut hole, into the hollow tunnel that runs through the center of your body, through the digestive tract, only then is it safe to activate them. Okay. Now, here's the thing that we were talking about today. So here we have the small intestine. And remember we talked about how the wall of the small intestine has all these finger-like infoldings. What's the purpose of all these infoldings?
More surface area.
Yeah, more surface area, which aids what process?
In the process of basically the absorption
Absorption,
of fat.
that's right.
Yeah.
Notice that these big projections are called villi.
Mm-hmm.
And then the small projections on the big projections are called microvilli.
Mm-hmm.
So this right here that I'm tracing is a villi, and then all these little things are the microvilli.
Okay.
And they're all operating here to increase the surface area. And now notice inside of those projections, we have the lacteal and the capillaries. So the food matter would be transported. The amino acids and the monosaccharides would move out of the lumen into the capillaries and into the bloodstream. And we saw how the fats would move out of the lumen. These are the epithelial cells. Remember that in the epithelial cells, the fats are turned into chylomicrons, and then they would be taken into the lacteal. And the lacteal is part of what system?
Lymphatic.
That's right. By the way, eventually, the lymphatic system eventually delivers things into the bloodstream. So eventually the fats do reach the bloodstream. First, the fats are taken into the lacteal, which is part of the lymphatic system, and then eventually it'll be delivered into the bloodstream. All right, so this is a better picture of what the villi and the microvilli look like. So here's the big thing we were talking about here. So in the lumen of small intestine, so another name for fat is a triglyceride. Another name for fat is a triglyceride. The triglycerides are broken down into fatty acids and glycerol, and also one other thing I didn't mention called monoglycerides. And then what do these arrows show happening here? Can you interpret what's happening in this picture? Take your time and try to understand what's happening in this picture. What does this show happening?
1 second, sorry.
Take your time.
Could you, what were you saying?
So this picture kind of summarizes some of the ideas we talked about for absorption of fats. So let's just make sure. So let's make sure we understand what's happening here. So what's happening with these arrows? What happens to these fatty acids and glycerol after they're formed in the lumen of the small intestine?
So the glycerol and fatty acids are absorbed. So that is the intestinal epithelial cells.
Right.
So they're absorbed into the epithelial cells.
Notice that's how it's labeled here as epithelial cell, and here's its microvilli. Good.
Yeah. And then
what happens to them once they're inside the epithelial cell?
The glycerol and the fatty acids are recombined into fats.
Right, which are also called triglycerides. Good.
Yeah. And then the fat is mixed with cholesterol, and then it's coated with proteins to form the chylomicrons.
Good.
And then that chylomicron becomes it's a water soluble globule, so it means it can it's water soluble now because fats are not usually water soluble.
Right.
So the chylomicrons move out of the epithelial cell and into the lacteal. Which the lacteal is a vessel in the lymphatic system.
Good. And as I was mentioning earlier, eventually it'll still get delivered into the bloodstream, but first it goes into the lacteal. This is maybe a clearer picture than I was drawing before, so hopefully this makes it clearer what's happening in the absorption of fats. Here's something we talked about. So what does this show?
Which one?
Can you see my cursor?
Oh, sorry. Yeah, now I see. Never mind. The hormones. Okay, so basically,
what's happening here?
The gastrin is being released when it senses that there is food in the stomach.
Good. And what does it cause the stomach to do?
It causes the stomach to release its gastric juices,
which contain
hydrochloric acid and pepsinogen, I mean.
Excellent. That's right. So that was one of the hormonal control things that we talked about together. Now, what's happening with this pathway?
This pathway is saying that how both CCK and secretin are being activated to release the bile from the gallbladder. And then CCK releases bile. It targets the bile in the gallbladder, and that allows it to be released.
Where was the CCK released from?
CCK is released from the small intestine.
Right. You can see that from this line coming from the small intestine. Good.
and then it also targets the pancreas, and that CCK allows the pancreas to release enzymes.
Good.
Mhm.
Good. And then what's this showing?
The secretin targets the pancreas, which secretes the bicarbonate
Good.
from the pancreas.
What's the function of the bicarbonate?
The bicarbonate is supposed to neutralize the wait, bicarbonate. I memorized it. I'm just forgetting it now. The bicarbonate is supposed to neutralize.
So it neutralizes the chyme that's entering into the small intestine. It neutralizes the hydrochloric acid that was coming from the stomach.
Yeah.
Okay.
Yes.
By the way, let's make sure we know what all these are. So what organ is this?
Stomach.
And what's this?
The esophagus.
Yes, the last part of the esophagus. And then what's this?
Small intestine.
Good. And this?
Gallbladder.
And this?
Liver.
Excellent. And this?
Pancreas.
All right. Okay. Good. And then what are they showing happening here?
The inhibition. Negative feedback from. It's basically saying that the food that just entered the small intestine, the secretin and CCK are both basically telling the stomach to slow down digestion so the small intestine can catch up.
Okay, good. And here's something that we talked about a lot. Okay, so notice here, when you eat, what happens to your blood glucose levels?
When you eat, it'll increase.
And then what's the response?
The response will be insulin being released
from
the liver.
Take your time. What does the chart say is happening?
Pancreas.
Yeah.
I'm trying not to
okay, yeah. Okay, so the pancreas would secrete insulin, and then...
the pancreas would secrete insulin, and then the glucose will become the glycogen.
And that happens where?
And that happens in the liver.
So this is a drawing of the liver.
And the muscles.
Good.
So this causes a decrease in blood glucose levels. Okay. The body cells take in the glucose. And the glucose is stored in the liver and the muscles.
Well, careful. Those are two different things. So the glucose is stored in the liver and the muscles in what form?
As glycogen.
Right. Now, in a separate path, the glucose is taken directly into body cells. Mm-hmm. And it's not changed into glycogen there. It's just maybe converted into ATP in glycolysis. So those are two different paths that they're showing here. And then that brings us back to homeostasis. On the other hand, what would happen if blood glucose levels dropped?
If blood glucose levels dropped, the pancreas will release glucagon.
So that's what they're showing here. Good.
The glucagon will move into the liver, and then the glycogen will be broken down into glucose and then released into the blood.
Okay, good. And that would reverse the original effect.
Mm-hmm.
Okay, so again, we went over a lot of material today. I wouldn't expect you to have it perfectly memorized yet. I don't know how much detail your instructor expects on these, but I tried to go over what I thought would be helpful on the test. I tried to talk about the stuff that I thought was most likely to be tested. Okay, so we went over the objectives here that I thought would be most useful. Obviously, it would be good to go through that was trickiest, and you could spend your own time to go over the other objectives. So I think we've covered the most important stuff here. And now would be a good time for you to just work on memorizing that on your own or filling in the gaps on your own. These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos at my website. There's a link to my website in the annotation. The address is www.freelance-teacher.com/videos.htm. Or you can just use the link in the annotation. Thank you.